Morning, everyone. Morning, class. So welcome to the Marine Conservation and Policy Program's annual symposium.
This is the 10th annual symposium.
10th annual symposium.
Although that
Thank
assumes
you.
didn't won the first year, I'm not sure. But let's grab it.
Okay. So this is the 10th anniversary. That's great. And I think most of you know who I am, but there may be a few of the incoming students I haven't met yet. I'm Ellen Pikitch. I direct the program, and I will be going on sabbatical as soon as this ends. And Anne McElroy, who is also wearing a blue mask today, will be the interim director, and she takes over the instant this is over. She'll do a great job because she really was the instigator, one of the instigators for this program, and has been involved with it from the very, very beginning. So I'm leaving you all in good hands, and I'm not gone yet. Okay, so before we go with the presentations, I hope you all have an agenda, and we'll be taking student presentations. Each student has 12 minutes to present, 3 minutes for questions and answers. We're going to keep to time so that everyone has a chance to present what they want to and get questions and answers. I'd like to invite our dean, Paul Shepson, to come up and just say a few words.
Do I know how to do that?
I don't know. I think so.
All right, so welcome to our new students. First, raise your hand if you're a new MCP student. Well, congratulations. This is a great program you've chosen wisely. The world needs you, the oceans need you, and I just want to thank you for being in this program. I'm going to just give you one piece of advice while you pursue your studies in ocean conservation. You'll be a student all your life and study broadly. The social sciences are incredibly important to ocean conservation, as witnessed as just one example by the fact that it is a matter of polite courtesy everywhere you go for someone to say, would you like some water? And they'll hand you a bottle, a plastic bottle this big. And the feeling is, I'm being nice to you, when in fact that little piece of plastic lives forever. Anyway, think and act and behave and pursue your scholarship broadly. To those who are finishing up, congratulations. I'm really looking forward to hearing your presentations, and you've done a great job. And all we expect for you is to go out and save the world. So good luck with that, and we'll be with you in that process. Thank you.
Thanks very much for those inspiring words. This is always my favorite part of the cycle. It's the day where our students who've been working for a whole year finish and tell us what they've been up to. And I think that for both current students and new students, it's great to see how far our students come in a year and the diversity of topics that they work on. It's pretty amazing, the lineup that we have for today, and it's always really exciting to me to see it. So for those of you who are entering the program, this is a sample of the kind of thing that you might do. And for those of you who are finishing, you'll get to see what your peers have been up to, in case you haven't been keeping close touch over the summer. I know you've all been out busy, some of you in the field or in the lab or behind a computer this summer getting these projects done. The program has had great success in its graduates getting jobs in the field of marine conservation and policy. And I got to really see that in the past year, where we took some extra special steps to try to connect with alumni, find out what they're doing, and hoping to continue to develop that network of alumni, to develop a sense of community, to keep it going. So I hope that this is not your last day here for those of you who are presenting, your last communication with us anyway. We hope that you'll stay connected and perhaps help the program in various ways. I know that at least one of the students this year was actually mentored by an alumni who is now with the Surfrider Foundation. So we are starting to see that kind of community going on. Okay, so any questions before I start? Okay, then I won't hold up the show. So we have a variety of presentation types. The first is going to be a presentation by Jason Chan, and he is here. So he'll be coming up to the podium and presenting in person, and then we'll switch to someone who is presenting remotely. Why don't you come up, Jason, and I'll be the timekeeper. Try to advance the slides. Great.
So, okay, let's start. So hi, my name is Jason, and my capstone topic is developing an indicator of marine heatwaves for the neophyte ecosystem.
You get closer to the mic.
Okay, so my advisors are Professor Janet Now and Professor Chris Wu. Where's my pictures? I have fish pictures to show you, but that's fine. So a little bit background about my project. So it all dates back to when I was an undergrad volunteering for Janet's lab on seaweed. Because Janet and other so much faculty were working on ecosystem indicator reports for the New York DEC. This indicator got really important decision for the neophyte and DEC. And one of the indicator is marine heat waves. So why marine heat waves? Because marine heat waves have a really overwhelming negative impacts on the marine ecosystem surface and health. For example, the 2012 Northeast marine heat wave and warming led to the decline of Atlantic cod and the lobster fishery. So what is a marine heat wave? So definition is when seawater temperature exceeds a seasonally varying threshold, usually the 90th percentile for at least 5 consecutive days, recommending using a 30 years long time methodology to be your average to reference to your heat wave. So here is the graph.
Jason, hold on a second. I'm gonna we'll try to move that bar down.
Oh,
so
yeah.
It's not totally in your way.
You can move that.
Yeah, you can move it around if it's on top of something you want to see.
Oh, okay. So where was I? Oh, back to the graph. So the blue line, so you have your temperature and time on your axis. The blue line will be your pathology, which is the average temperature that you want to make reference to for a heat wave. So the green line there, it will be your 90% threshold. The squiggly black squiggly line will be your temperature. And if your temperature goes beyond up that green line, that means for at least 5 days, that means you have a heat wave. So there are 3 types of heat wave statistics for the indicator that pick. One is humidity intensity, which is the red area over there. It's the integration over temperature and time. Second one is longest heat wave that has the longest duration. And number 3 is the number of heat wave events. Next, come on. Stop going.
No. Just, I think if you just click on the, that's not working.
That's what Mark gave us.
Oh,
Oh,
all right.
okay.
Or you
Thank
can,
you.
or you can click on this.
Yeah, I think you said though. That's what Mark gave us.
Okay. So some of the things that I learned from my background reading is that the Northeast shelf is warming like really fast.
Excuse me,
So-
Jason. It'll really help us if you stand as close to that microphone
Oh.
as you possibly can.
Okay. Sorry. So, okay, can you hear me? Okay, nice. So the Novi Shell is warming pretty fast. For example, the warming in the Gulf of Maine increased faster than 90% of the global ocean in the past decade. And we are seeing more frequent, longer and stronger marine heat waves that seems to be correlated timely with the past decade warming of the Novi Shell.
Is there?
Oh, what is it?
Okay.
Okay. Oh, okay. So here are my hypotheses or questions that beat through my head. So I think the art of detecting marine heat waves depends on the climatology selection, which is how you pick your average that you want to make reference to the heat wave. And they recommend a 30 years long climatology, but I think I would question if that worked well for the Novi Shell. Would not be, and one thing is that considering the increase in temperature, it would not be sensible to pick a long climatology that covers a long period of warming, and it moves up your threshold. But too short climatology may not be a good representation of a good average as well. So the ultimate question is that what is the best climatology? So I have hypothesized that using different lengths of climatology from different periods may change the result of my heat wave detection, and then affect these heat wave indicators and management decisions. So the material that I use the OISST from NOAA. Is a satellite data set from 1982 to 2019, covering from the Mid-Atlantic Bight to Gulf of Maine. So for the detection, I use MATLAB detection, heat wave detection package. I also use R, Excel, and GIS as well. So first, what did I touch? Oh, anyways. So first, I create a list of 25 different climatologies with different lengths and from different periods. Oops. It's not touching.
No, it's not a slide. Go back.
Oh, nice.
There we go.
Okay.
It's on it.
So, okay, so I first create a list of 25 different climatologies from different lengths and different periods of my 38 years of OIS temperature data. And here is the list. So here is the list. So you can see that there are 25 of them. How I create them is treat my data set as a timeline, and then I cut them into 5-year chunks. And then I just sort of grab chunks of data to be of different lengths from different periods as these 25 different climatologies. So first thing to do is to run the package using these 25 climatologies. So test water to test for the 2012 marine heat wave for cumulative intensity. Then I do a t-test to see if these detection results are significantly different. And then I'll run these 25 different climatologies year by year from 82 to 2019 and calculate 3 types of the marine heat wave statistic that I mentioned before. And then I graph all of them to see what they look like. And compared to the standard that in the green dots. Actually, the standard is the one they use in the UO5 report. So it can be my reference. So here is the t-test result. It's a pairwise comparison table that I highlight all of these p-values in red that are less than 0.05, which is the alpha value. That means most of these climatologies produce very different results. And it gives me the purpose or the encouragement to move on to the next step, because when you use different climatology, you produce different results. So here are the graphs. It looks really messy. So I break it down for you slowly, step by step. So the way I read it is to first look at the big red line there is the standard climatology, and all these color lines are the climatologies. So when I talk about variations, I'm referring to the gaps between those climatologies. So looking at cumulative intensity and the longest marine heat waves, right? On the top and the middle graph there. So after 2007, different climatologies show that increase the variation to detect marine heat waves. Some of them may over-detect and some of them may under-detect heat waves during these warm years. So before 2007, number of marine heat waves detected, which is at the bottom, they show really consistent variation compared to the standard. But after the warming in the 2012, they all systemically under-detected the marine heat waves compared to the standard. So for all climatologies, right? So for all the climatologies, they saw an increase of variation to detect heat waves in the years with more heat waves. Also vice versa. Come on. So here is actually the same, same figure from the last slide. I just pulled some of data out so it's easier to see. So the medium climatologies are the one that around 20 years, we begin to show signs of variation in detection sensitivity. Some of them may over-detect and some of them under-detect heat waves compared to the standard. So the one that over-detect is the one that over the red line. The one that's under-detect is under that red line. So here's the map. You can see that one is detect a lot of heat waves, and another one doesn't detect a lot of heat waves. They are the same names, but from different periods. So for the shorter climatologies that are less than 15 years or so, you can see there are more variation in the heat wave detection sensitivity. You can see that more under detection and more over detections. Sensitivity also depends on if the climatologies come from different periods, like I said before, different chunk of my data set. So it's the map, one is more, one is less, like the last one. That little one over there is the standard. Yeah, it's the standard one. So for the really long climatologies that are 25 years or above, covering recent decades warming, tend to have the least variation among the three types of marine heat wave statistics. So you can see those color lines, they are kind of really packed together, really tight together, unless compared to the previous one that has really big variations. So these kind of climatologies tend to be less sensitive to marine heat wave compared to the standard. So here's the map. They don't have a really big difference, so less variation to detect these marine heat waves. So here is my summary table for the things that I did. For everything, these detection sensitivity compared to the standard is really matters, because the one that in red, that means it's under detecting. Okay, my conclusion. Okay, climatologies that covering recent decade warming, regardless of length, are gradually less sensitive to heat waves compared to the standard 30 years. Long climatologies that are more than 25 years to cover recent decade shows that the best consistency in heat wave detection performance. So it's due to the past warming, past decade warming. Most of the climatology and the threshold warming in the low threshold in the past decades is enough to affect the result of heat wave detection, regardless on how you pick your climatology. The best is to avoid climatologies that is covering recent decade, but still have a reasonable long length. So I'll say descendant is a pretty good one because it has a nice balance on both. So that's all.
2 minutes for questions
Oh
now.
yeah, I'll skip that for the question.
Okay.
And next steps, if that, okay, thank you.
Okay.
Oh, yeah. Speaking too fast, keep getting along.
We have time for a question or two, depending on how long they are.
Okay.
You had two spikes, I think it was 2012 and about 2015. Is already evidence of the biota responding to those.
Oh, yeah. So that's what I'm doing. The planning to do the next step is to generally, of course, to connect that with fish data sets and see if that detection how it changes the correlation between those fish distribution abundance and the central biomass. Yeah.
Okay, maybe a quick question.
Because we are seeing more sea bass and flounder during the year of more picnics in the Norfolk Shore.
Great, thank you.
Yeah, thank you.
So our next speaker is Amy Bavosa. Amy was going to be presenting in person, but yesterday she tested positive for COVID. So she decided that it would not be a good idea to come and present in person. So she will be presenting virtually. She said she's okay, well enough to present. I obviously gave her the option of putting this off to another time. Amy, are you ready?
Yes, I'm ready.
Great. I'm so glad that you can join us.
Me too.
This
I
is-
am vaccinated, by the way, just somehow got COVID. So here we go. Let me get this into the right format.
Okay, I'm gonna hit.
Okay, you can see that.
Yeah.
Okay, so I will be presenting on Porites, astreoides, and Neutrogena sunscreen, the effects of UV filters on coral reproduction. So coral reefs cover approximately 2% of the ocean, but support about 25% of all known marine species, which includes roughly 1 million species of fish, invertebrates, and algae. Coral reefs also provide goods upon which more than 500 million people rely upon by supplying necessary sources of food through fisheries, generating local income through tourism industries, and providing barrier storm protection. Unfortunately, however, coral reefs are experiencing a significant decline, as we all may know. So some threats to coral reefs. Current threats to coral reefs include warming seawater temperatures, ocean acidification, overfishing, and damage or destruction from coastal development. Within the last 40 years, it is estimated that there has been a 20 to 50% loss of coral reefs. And while chemical stressors on spawning corals are not yet widely understood, the dangers of UV filters in sunscreen are starting to come into the spotlight. So UV filters, they're found in both personal care products and sunscreens and used to protect users from UVA and UVB rays. So while we know that they're used in sunscreens, they're also found in some items that you may not suspect, like shampoos, fragrances, and even plastics. They can be either organic, which is carbon-based, or inorganic, mineral-based. And NOAA has identified some harmful chemicals in sunscreen, though not all of them, to include oxybenzone, benzophenone 1, benzophenone 8, ODPBA, 4-methylbenzaldene camphor, 3-benzaldilene camphor, octinoxate, nanozinc camphor, and oxide, and octocrylene. So UV filters and water quality. Once UV filters enter waterways, they can induce bleaching in corals, alter their DNA, deform young, and possibly even kill corals. It's estimated that between 6,000 and 14,000 tons of sunscreen lotion, many of which contain between 1% and 10% oxybenzone, are released onto coral reefs each year. And the US Center for Disease Control estimates approximately 96.8% of the US population is exposed to oxybenzone. And part of the reason why these are so problematic is that UV filters are not easily removed by wastewater treatment plants. So they're not only a risk from people putting on sunscreen, going into the ocean, and then washing off there, but also being washed away in the shower or down the sink. because wastewater treatment plants are designed to remove larger particulates from the water. So it's estimated that about 10% of global reefs are at risk and 40% of coastal reefs are at risk. So some more effects of UV filters. UV filters don't affect just coral. UV filters have been identified in various fish around the world, leaving further implications for the food chain. UV filters impair photosynthesis in green algae. They can induce defects in the young of mussels and sea urchins. UV filters can decrease fertility in fish. They can accumulate in the tissues of dolphins, and they have even been found in the Arctic. So it's a much larger problem than just, isolated tropical areas where the coral reefs are. So there have been some movements to ban UV filters and the harmful chemicals. Policymakers in places such as Palau, the US Virgin Islands, and Hawaii have successfully banned certain UV filter compounds from being sold. However, in places like the Florida Keys, there was an initial movement to ban UV filters, but the law was not upheld based on the fact that they didn't think there was enough evidence to support the ban. And then some lawmakers also questioned the effectiveness of this ban. Maybe there's another route that we should be taking, education outreach, promoting alternate forms of sun protection. So the study that I did took place in the Florida Keys at Mote Marine Laboratories Center for Coral Reef Research and Restoration. As our sunscreen, we chose to use Neutrogena Ultra Dry Touch Sunscreen with SPF 55, which contains 5 UV filters: homosalate, oxybenzone, octisalate, octoprilene, and avobenzone. We decided to use the entire sunscreen for this study rather than just an isolated UV filter, so that we could improve our chances of results. We figured that later on, based on the results, we could always do another study with the isolated compound. We use Porites astreoides coral since they're a hardy species and release fully formed larvae, which allow the opportunity to observe both the adult coral as well as the larvae. And in the Florida Keys, the larval release occurs in April and May during the full moon, so it's kind of easy to track their cycle. And the motivation for this study was to investigate the impact of environmentally relevant levels of oxybenzone and other UV filters on Porites spawning in order to help inform decision-making. So the methods. The study started April 9th and concluded a little early on May 15th. We collected, or the dive team actually collected, 40 intact Porites colonies from nearby, and they were randomly assigned to one of 6 flow-through tanks. Each tank was outfitted with 2 pumps to keep water circulating within it, and the water flow into the tanks was calibrated daily to ensure that 1 liter per minute was flowing through and also therefore out of the tank. Of the 6 tanks, 2 contained controls, 2 contained corals exposed to a low dose of sunscreen, and 2 contained corals exposed to a high dose of sunscreen. To initiate the study, 6 milliliters of sunscreen solution were mixed into the low dose tanks and 60 milliliters were mixed into the high dose tanks. After that, peristaltic pumps continually moved sunscreen solution from a low dose and high dose reservoirs into the tanks at 1 milliliters per minute. So to make the sunscreen stocks and solutions, there was an initial sunscreen stock made from 60 grams of sunscreen mixed into 300 milliliters of autoclaved seawater. The low dose mixture was made of 0.8 milliliters of concentrated stock into 3,999.2 milliliters of seawater. And the high dose stock was made from 8 milliliters of concentrated stock into 3,992 milliliters of seawater. And the high and low dose solution had to be made every 2 days. And these were also vigorously shaken before we connected them to the peristaltic pump. And they were also continuously stirred by magnetic stir bars and stir plates. Other daily tasks for the study included feeding the coral, cleaning the tanks, taking physical parameters such as dissolved oxygen, water temperature, salinity, and pH with a YSI. And we also did solid phase extractions about once a week so that we would be able to determine the concentration of UV filters in each tank. So here you can see our spawning setup, which was different from the exposure tank, and then some of the Porites larvae. So for the spawning cycle, we had to move the corals from their tank to an indoor raceway where the collection was set up. Here you don't see the corals in the bowls, but Each coral was placed into a small mixing bowl that had a handle that led into a small plastic beaker that had a mesh bottom. So there was water continuously flowing through it. And they each had an individual hose going into each bowl so that there was also continuous water flow there. In the morning after they spawned, we would, it was really exciting to come in and see all the baby larvae. And we would collect them using pipettes and put them into jars respective to each coral that had spawned. Once they were collected, we counted the individual larvae and put them into settlement plates and also froze some for later analysis in the lab. Those are results that we're still waiting for. And for the settlement plates, we watched settlement over the course of 5 days. So we have a few results, not much that we can look at from this study. So during the cycle, we had two cycles of reproduction, the first full moon and the second full moon. During the first full moon, over the course of 2 days, the control corals produced an average of 234 larvae. The low corals produced an average of 261 larvae, and the highs produced an average of 179 larvae. The numbers were drastically different on the second full moon. We're still not sure why exactly. On the second full moon cycle, the controls produced an average of only 8 larvae, the low produced an average of only 11 larvae, and the high produced an average of only 3 larvae. And then in terms of settlement, I chose to look at them as percent of the total larvae that were put into settlement trays. So on the first cycle, 44% of control coral larvae settled, 54% of the low larvae settled, and 55% of the high dose larvae settled. On the second full moon, we were working with much smaller numbers, but still 11% of the control coral larvae settled, 36% of the low dose coral larvae settled, and 58% of the high dose coral larvae settled. And some conclusions. Though our results are currently inconclusive, further work in the lab with the larvae may reveal adverse effects in areas such as size, protein content, and symbiont density. This experiment was an important step towards making replicable studies that could give more weight to bans on sunscreen with harmful UV filters, since it was often cited that studies weren't replicable or reputable. And labels matter. Currently, there's no sort of advisory board on who gets to say if sunscreen is reef safe or not. So even sunscreen with harmful chemicals are able to advertise as reef safe. So be sure to check the backs of bottles if they contain chemicals such as oxybenzone or homosalate. And we must continue to use outreach, education, and awareness, which can play a critical role in keeping corals safer. And that's it.
Great job, Amy.
Thank you.
That's a little depressing, though. That's even things that say reef safe may not be. We have a lot of problems to fix. So there's plenty of time for questions. Anne is dying to ask a question.
Well, Amy, let me congratulate you. This is, it's so important to actually evaluate these kinds of things, and working with the real stuff, what people put on their bodies is probably the way to go, because that's what getting off into the water. Also, let me congratulate you on actually doing a full-scale pilot study. You know, even though you didn't have the statistics to show anything definitively, you obviously learned a lot from this study, and this will guide, you know, the follow-up. Hopefully, there will be one that really hopes to nail this. And I was just listening to NPR on the way in today about interpreting data. They were talking about incidence data of people vaccinated who were getting COVID. It totally speaks to the situation here. But you know, particularly with larvae and larvae settling, often you don't have high numbers. I mean, it's a dangerous thing being a larvae settling in the world. Most of them don't make it. That's the norm. Chemicals are not, I think. And so, you know, you might consider looking just at total of you know, the percentage, that not getting away from percentages and looking at total numbers, because sometimes it gives you a different, you know, different endpoint. You know, if you could just up the replication of this and repeat it, you might have a really excellent study here. So well done. I guess that was.
Thank you.
Great. We have some time for another question. The dean would like to address you.
Always do it. So yeah, really nice job. It's very interesting. Sort of thinking from the perspective of reviewers or the manufacturer of the sunscreen. When looking at the data for significance, I would want to see the actual concentrations of those compounds in the water. And the reason I say that is just as an example that tubing, I don't know if it was Tygon or whatever it is, these organic compounds would be very, to use a technical term, tubingophilic. They would be soluble and absorb on the tubing. And so you never know what you have in the actual test water unless you measure it. I mean, here we could get help from, LCMS, from the Center for Clean Water Technology or someone like that. Maybe you did that, but it's really important to actually analyze the water.
So we did do solid phase extractions, which was taking a water sample and then sort of extracting down to the compound to see how much of it was in there. We just didn't have those results ready yet.
Oh, okay, great, excellent.
Yeah.
Another very quick question. Glenn.
You had big differences in the number of larvae spawned at the two spawning events. Were the corals in the second spawning event, do you know whether they had already spawned once or not? Do you know their spawning history?
Sorry, I'm having a little bit of trouble hearing you. I think the question was.
What Glenn was asking is in the second spawning event, actually, help me.
Were those individual corals that spawned in the second event, had they already spawned?
Okay.
And does she know that?
Okay.
Some of the corals that spawned were repeat spawners, but not all of them. Some of them had spawned for the first time.
Thank you.
Great, and we're exactly out of time. Thank you again, and
Thank you.
congratulations. So next, we have Amanda Hanifin, who is here. So now to Amanda.
Hi, everyone. For my internship this summer, I did a dual internship working with the Shinnecock Bay Restoration Program. I will be referring to that as SHARP from now on. And I also did a policy audit of best shellfish management practices in New York State with Pew. So Shinnecock Bay is a bay on the southern border of Long Island. And over the years, scientists have noticed that the water quality has been decreasing in the bay. Scientists have seen increasing temperatures, increasing pH levels. Sorry about that. Okay, so yeah, we've noticed the loss of biodiversity. Like I said, the eelgrass beds are decreasing, and so is the amount of fish populations, and especially shellfish populations in the bay. So in this case, restoration is key to be able to preserve the bay. And that's why SHARP was formed in 2012 by the SOMOS faculty, so that they can start improving the bay. And they needed to restore shellfish there because shellfish are filter feeders. So what they can do is that means that they can filter the water through them and remove certain toxins and pollutants from the water. Okay, so part of my internship was I went out trawling with the SHARP crew. So we do this so that we can monitor the bay and sample it. And at 13 different sites in Shinnecock Bay, you can see it on the map there, we took water quality samples at every single one using a YSI, which that takes the salinity, the dissolved oxygen content, and the temperature of the water. And at 11 of these different sites, we trawled, and this involves deploying a large net behind the boat and catching any type of fish or macroalgae in its wake. So at most of these sites, we would trawl for 3 minutes, but then at 2 sites in the middle of the bays, we would trawl for 7 minutes. And everything was brought up on board. We were able to identify all of the algae and weigh it. So we have we can take a look at the types of macroalgae in that area. And we also quantified and measured and sexed, if it was possible, all of the different types of crabs, fish that came up on board. And afterwards, they were safely released back into the bay. So I want to show you a video of what the trawling looked like, just so you can get an idea of what we were doing. So the guys, he's going to be throwing the net into the water, and then it'll show how the net is deployed into the bay. So they do that pulling motion there to help spread the net out into the water. And then it's released. And then once it's fully out, we start the timer, and it goes for however long we wanted to trawl for that at that site. So I wanted to show some examples of the types of algae and species that we found. In the upper left-hand corner, that's a type of ulva. We caught mostly ulva when we were out there. The next one is blue crabs. We caught a lot of sardines. There was one trawl, I think, at one site that they found. They caught over 550 sardines in that, so they had to count all of them. There's different types of flounder. The middle lower is the black sea bass, and then there's a pipefish. And horseshoe crabs, we found lady crabs, shrimp, pufferfish, skates, seahorses, and dogfish. And one that I did not include that I wanted to, but I didn't get a picture in time, was we caught, the last trawl was this past Tuesday, and we caught a mantis shrimp, and they haven't been seen in, I think, over 3 years. So this year was the first year that we were able to catch them again, and they're a good indicator species of a healthy ecosystem. So that shows that everything that SHRP has been doing for the bay is starting to work. So when we were out sampling once a month, we would take we would take water samples so that we could do eDNA analysis, and eDNA stands for environmental DNA. And this is the process of sampling the environment to obtain DNA in the form of like skin cells, scales, excreted substances. And so we would collect this water so that we were able to tell what types of species are in the environment that maybe we weren't sampling when we were trawling. So once we collected the water, we would bring it into the lab, and it consisted of 2 days. So the first day was filtering the samples through a filtration system. And we would filter 1,000 milliliters of water through a filter that catches all of the particles and DNA that's in these samples. And once it's done filtering through, some of them can take a long time. Some samples have been taken up to hours to filter through at some point. So it's a whole day process. So once it's finished filtering, we take the filter, cut it up, and put it into labeled vials so we know which filter is from which station. And we put them in the freezer overnight for DNA extraction the following day. And this is when certain buffers and solution are used to wash the filters. So they are washing the DNA from the filter. It goes through certain incubation periods, and their vortex are like heavily mixed so that everything can get thoroughly washed, and all of the DNA will be released from the filter and into a solution. And once we finally get that solution ready, it is, we use a nanodrop-like spectrophotometer to assess the purity and concentration. So the purity looks at if the solutions are contaminated in any way, and the concentration shows how much DNA is actually in the solution. So once we are able to do that, the samples are collected and then sent to a DNA processing lab called Mr. DNA, and they are able to actually determine the type of species that are present in the samples. So we can see if there's any type of fish or any other biomass that we might have missed that is present in the water. So SHERP has had a lot of success over the years. The SOMOS faculty has worked together to achieve the restoration goals of decreasing harmful algal blooms, increasing biodiversity of plants and animals, and restoring nutrient balance to the water. So part of what they did was they established spawner sanctuaries in the bay. And this is where a large number of hard clams are planted and no harvesting is allowed to take place in the area so that they can just grow. And they were over 3.5 million hard clams planted, and their harvest has increased by over 1,000% over the years. They also were able to construct 6 oyster reefs throughout the bay and spread over 5 million eelgrass seeds. So you have about 3 minutes. Okay, all right, thank you. So the monitoring continues, and eDNA is continued to use to find the species. So the last part and second part of my internship I began in January. I worked with Christine Santora and Aaron Kornbluth from Pew, and we basically compiled an shellfish policy audit that's still being worked on. It's not complete yet, of all the laws, regulations in New York State that has to do with shellfish restoration. And so it was originally over 150 pages, but we're cutting it down and going to be submitting it to Pew soon. That's everything. Yes.
So we do have time. Go ahead.
Okay.
Nice presentation.
Thank you.
So what's the most interesting thing you learned from the eDNA?
So I think the most interesting thing was that, well, I didn't even know that it had existed before starting it. So the fact that we can actually use eDNA to go out there and sample is really important because it doesn't disturb the ecosystem. You know, we can take water samples, and it's super important for the future because we do know that trawling can do some damage, right? So I think that it's such an important tool for future bay restoration.
But do you have any results from it yet to know what species are coming up?
No, we didn't get the results yet from what we did over the summer. Yeah, hopefully soon. Yes.
I know in a few classes we learned stuff like trawling is really bad. But you find is there any way you guys to make sure you do this safely?
I'm honestly I'm not entirely sure. I know that we only do it in specific areas and we keep it to the absolute minimum possible, but we need to do it just to see for now until we can maybe use eDNA totally to sample around and, you know, like for restoration work. Yes.
So I know you mentioned that it's an ongoing project. You're looking at the policies for protecting shellfish.
Mhm.
So I just wonder, what time frame are you looking at? And you said you have a preliminary document with your few collaborators. Have you seen any trends in throughout time that you're looking in terms of policies for that type of protection?
So we started this audit back in January. So I'm looking at the most recent laws and regulations. I haven't looked at any in the past. So they're present day laws, and we're hoping to get it done by September. We're almost finished. We only have a little while left to go, but some other things came up with my advisor, but we're continuing to work on it. Hopefully it'll be done soon.
Sorry.
No, that's fine. Thank you.
Okay, thank you very much. So a couple of things before our next speaker, who's going to be remote. So Amanda, when she answered the question about do you have any results, she said not yet. That means for 2021. We do have results from 2020 and some preliminary results from 2019. Very pleased with the results. I don't want to take up the symposium to talk about it. The other thing on the New York Shellfish audit, we got a grant from Pew to start that work and to create an inventory. We're now working on a proposal for the next phase, which would be to have a workshop, bringing together all the players of interest, who have an interest in this in New York State, to develop that, take that inventory and turn it into best practices recommendations. Okay, so next is Camilla Piechocki.
Right.
Hey guys.
There.
Right,
Go ahead.
Cam, you want to share your screen and present?
Fantastic.
Sure.
Okay, and
Great.
so this is a very creative and appropriate title for this talk.
Thank you.
Go ahead.
So good morning, everyone. My name is Camilla Piechocki, and welcome to my presentation entitled A Whelk-coming Alternative to Using Horseshoe Crabs as Bait. So we'll start with a brief outline of what I'm going to cover in this discussion. We'll start off with an introduction to the two major players, channeled whelks and horseshoe crabs, before going into the research questions. From now on, whenever I say whelks, I am referring specifically to channeled whelks, and the abbreviation HSC refers to horseshoe crabs. I will go over the methods used in each location and discuss the type of data collected and how it was analyzed. Then I'll explain how this capstone project relates to marine conservation overall. And whenever I talk about this project, the most common question I got was, what exactly is a whelk? Well, basically, it's a sea snail. It's in the phylum Mollusca and in the class Gastropoda. Channeled whelk actually grow quite slowly, and it takes them years to sexually mature, which leaves them vulnerable to overfishing. This is a growing concern because the whelk fishery has existed for over 100 years. However, in recent decades, there has been a growing demand in Asian markets. And so every spring and fall, which are near the top, whelks are near the top of the list for fishermen. But to catch whelks, fishermen have learned that they first need to catch horseshoe crabs. Despite the name, they are not actually crustaceans, but instead are chelicerates. They are an important source of food for endangered shorebirds that rely on the nutrients from their eggs to be able to migrate to their own nesting grounds. However, the ASMFC lists horseshoe crabs as poor status, while the IUCN Red List labels them as vulnerable. The reason why they are in danger is primarily because of overharvesting by fishermen to be used as bait for catching whelk. However, there is one recent solution that does sound promising. And this solution was created by Kepley Biosystems Incorporated. They used amino acids to build what looks like a hockey puck or a powder that smells like shellfish and decaying fishes, such as menhaden, which whelks are known to eat in the wild. Since this alternative bait smells like their natural prey, it makes sense that they would be attracted to it. Interestingly enough, although they seem to be attracted to the scent of horseshoe crabs, crabs are not known to be part of the channeled whelk's natural diet, and scientists are not quite sure where the attraction comes from. Kepley Biosystems collaborated with Cornell Cooperative Extension for this project. Fortunately, since the alternative bait is completely made in the lab, it does not require any horseshoe crab and is a more sustainable alternative. In theory, whelks should be eating this up. But prior to this experiment, they haven't been properly tested. So I worked with my advisor, Dr. Matt Sclafani from the CCE, to test out this alternative bait. And together we came up with two main research questions that we wanted to answer. Firstly, are whelks attracted to organobait? And secondly, do whelks prefer organobait to female horseshoe crabs? And we chose female crabs because that's what fishermen have traditionally used. Additionally, I decided to look at parasite loads to see if those could affect behavior. To accomplish this, we had to set up a choice arena, which you can see in the bottom left corner. But before we could do that, we had to set up a home for our whelks, which were donated to us by friendly fishermen who caught them as bycatch. I actually set up surveillance cameras for the first few whelks in the holding tanks that we received so that we could observe what times they were active and therefore learn when to schedule the trials. A screenshot from that is in the lower left. I also set up a feeding schedule to ensure that whelks were starved for at least 3 days before the trial began, so that they would be enticed by the smell of food. Dissections were also done in this location, but in a separate lab. Meanwhile, the experiments themselves were done in a choice arena that used a design based on Jutfeld et al. 2017, and we adjusted the size to be able to be used for whelks. The key thing to note here is that this setup had to maintain laminar flow. So water would be pumped from the barrel in the red circle, carrying 1 cent through the pipes in the purple rectangle. This flume would flow through a collimator system that would straighten out the flow and prevent mixing. Meanwhile, the water in the barrel in the blue circle carried a different scent. The whelk's activity was captured by a camera mounted on the ceiling, which you can see on the right. I managed to run 4 successful control trials with organobait on the red side and plain seawater on the blue, and 4 experimental trials with organobait on the red side versus horseshoe crabs in the blue. Each trial took about 2 hours to run. And to give you an idea of why, I like to show this quick clip of the fastest whelk, Grease Lightning, which had the highest acceleration of any whelk at 1.19 centimeters squared per second. So we had to give them some time to do their thing. And the videos that we got from the ceiling camera were analyzed using version 5 of the LowlyTrack software. It measured each snail as a blob and tracked their movements, and even created heat maps of concentrated activity. These maps can be important in understanding the numbers, because as you can see in the upper right corner, Grease's acceleration occurred primarily in the organobait zone. To analyze the overall amount of time spent in each zone, we ran a binomial proportion test, and t-tests were run to test for significance in acceleration and speed. Despite a myriad of logistical challenges, because this hasn't been done with whelks before, we got some interesting results. On the right, you can see 2 graphs depicting the percent of time spent in each zone. In the top, there is a noteworthy preference for organobait over seawater in the control trials. And in the bottom, there is a clear preference for horseshoe crab over organobait. Both of these are statistically significant and give us a clear idea of where the whelks wanted to spend their time. A subsample of the whelks were dissected, but no parasites were found in the gonads or hearts. Now, on these graphs depicting average speed and acceleration, none of the p-values are statistically significant, which is most likely due to not having many replicates. Remember, we were only able to run 4 successful trials within the control and experimental. So despite the small, the number of replicates was probably the strongest limiting factor for this project. But there are some tendencies in the control trials to be more active in the organobait zone. So the higher rates of speed and acceleration could suggest interest in the scent. So overall, whelks definitely preferred smelling organobait over plain seawater. But when it came down to it, they still preferred horseshoe crabs more. So despite the small sample sizes, this experiment showed that there is promise for an alternative bait, and Kepley Biosystems is already considering new formulations. What's nice is that now we have a better understanding of how whelks function as laboratory animals, and a solid foundation to run future trials and address additional questions. For instance, we've come up with new hypotheses to test out in the future, such as, do whelks have a preference for menhaden in the fall and perhaps prefer horseshoe crabs in the spring pre-reproduction. And perhaps they prefer females over male horseshoe crabs because they're interested in the scent of their eggs. No scientific literature has yet to test pure horseshoe crab eggs as an attractant for whelks, so it would be really interesting to see. If someone does, then Kepley could definitely use that to tweak their formula. I'm glad there is promise in organobait because it's easier for fishermen to use. Instead of freezing an entire horseshoe crab, they just have to deal with a little hockey puck sized powder. Plus, if fishermen have another option as bait, there should be a decrease in illegal horseshoe crab harvests locally and potentially along the eastern coast. There are several agencies in many states that are interested in developing an alternative bait, such as the DEC here in New York. This will hopefully relieve some of the pressure on the horseshoe crab fishery and allow them to increase their abundance. This could be a game changer for horseshoe crab conservation. Now, really quickly, I'd just like to say thank you so much to everyone who contributed to this project, most especially my amazing advisor, Dr. Matt Sclafani, who was incredibly optimistic and encouraging. And if anyone has any questions or something they need clarified, feel free to ask.
Excellent job, Cam, and you left plenty of time for questions. Okay, thank you.
So I've heard of a different alternative that's called Biondi that just like assists keep the horseshoe crab. Do you know if that's preferable to whelks or did you not?
Were you able to hear that?
Something about biobait. Could you please repeat the question?
Yeah, an alternative. She was asking about an alternative bait that she's heard of called biobait. And do you know how it compares to organobait?
I'm not familiar with biobait, but I am aware of other options for alternative bait for whelks that do include horseshoe crab in their formula. And the main distinguishing factor with organobait is that it's trying to use no horseshoe crab whatsoever. So I'm not quite sure how it compares.
Cam, what about the costs of the fishermen, organobait versus horseshoe crab?
Were you able to hear that?
No, I'm sorry.
Okay, Dr. Taylor asked, what about the cost to the fishermen? Is horseshoe crab less expensive than organobait or vice versa?
So fishermen generally collect the horseshoe crabs by themselves. Sometimes they do enlist people to help them, which could be a cost to them. Generally speaking, it's more a cost of time and effort, more so than money. But organobait would just be a cost of money instead of that time and effort. So it would save them a lot of time, and some fishermen would definitely prefer that. We can't be 100% certain that fishermen would completely stop harvesting horseshoe crabs, but it definitely would be good to have an alternative option out there because we're certain that some fishermen definitely would rather sit on the couch at home instead of going out at night in the summer to collect them.
There's another question about.
Actually, I just want to add to that. One of the additional costs that's really the most expensive part of the bait is storing them in a freezer. The cost of operating a freezer year round. Purchasing one is not inexpensive at all. So it'd be cheaper than that. And much easier to pull something off the shelf.
Right,
Yeah.
so what was being said, in case you couldn't hear it, is that the expense to the fishermen is keeping the bait frozen. What about the cost to the ecosystem and the horseshoe crabs? Why are we so worried about horseshoe crabs, Cam?
We're worried about them because they do play a very important role in the environment. Their eggs are the major source of nutrition for endangered migratory shorebirds. There's a few different species that rely on their eggs in the summertime, and they load up on those eggs to be able to fly and migrate to make their own eggs. So without horseshoe crabs, a lot of animals actually would go without a vital food source, and you would definitely see a direct impact in the populations of shorebirds.
And what about the life history of the horseshoe crab?
The
Does it,
life history is, I'm sorry?
does it make them vulnerable to overexploitation?
Oh, definitely. Horseshoe crabs take about 10 or 11 years to reach sexual maturity. And not a lot of juveniles survive until that age. So it takes them a very long time to be able to repopulate. And the fact that they are being overharvested is what has led them to become, to be listed as vulnerable by the IUCN Red List. So there are different companies with different approaches that are trying to save horseshoe crabs and monitor their populations to make sure that they don't become even more endangered.
Okay, those were great answers. You left plenty of time to be grilled. One more. Carl Safino?
Anybody planning to do a field trial to just see whether you would catch as much with the artificial bait, even though in the lab they would prefer the horseshoe crab?
Did you hear that?
That is a great question. Yeah, I actually heard that one. Thanks, Carl. That's a great question. I didn't think I'd actually have time to mention the field experiments, but there were some preliminary field tests done, and they actually showed similar results to the lab experiments in that in the areas where it was organobait versus an empty trap, there was definitely some preference for organobait, but when there was an organobait trap near a trap with horseshoe crab in it, they still strongly preferred the horseshoe crab. But those were preliminary, and I didn't think I'd have time to talk about them.
Okay, well, thanks again. Congratulations.
Thank you.
And our next speaker will also be presenting remotely, Chris Biondi. Oh, look, he started with this.
Oh, yeah, quick.
Oh, Chris, good job.
Hi, everybody. I'm Chris. I'm going to be presenting a project called Conserving the Value of Time, Identifying Mature Forests in Orange County. I want to quickly thank both my advisors, Robert Serrato and Matt Decker. So my number one goal for this project was to identify mature forests on a large scale. So we all know that, counting the rings of trees can help determine the age of a forest, but doing that for over a large scale area is almost impossible. But with spatial data, and it can give you an accurate assessment of forest age without actually visiting each forest. And the second goal of my project was to integrate my finished data set into the local land trust conservation plan. So this project was heavily GIS based. So those who don't know what GIS is or haven't used it, it's a software that allows you to capture and analyze spatial and geographic data. So why should we conserve mature forests over the younger counterpart? Well, mature forests are key in their carbon dioxide removal capabilities. And the older the trees are, the better they are at this. Existing trees have a higher carbon sequestration rate than newly planted ones. These forests also have higher levels of biodiversity and a higher rate of native species. They're also resilient against the changing environment, and large forests play a big role in water filtration and purification. Additionally, mature forests can be used for education and recreational activities. So the first step of my project was to find an existing source of historical forest data. So the first place, or the best place I found, was historical topographic quadrangle maps. So here on the right, you can see this is just one of 19 maps that would make up Orange County, New York. So you can see that this map was actually prepared under the US Army during World War II. I think it was a domestic mapping program that they were undergoing during that time. And you can see that all this green area is forest, and I think 1940s was around the first time they started putting color into these types of maps. So that makes it perfect for what I was trying to do. And this is actually a mosaic of all those 19 of those maps together into one, and this is Orange County. You can see that there are some differences between the maps. They're not all made by the same person exactly, so there's some color differences, but overall, you can still get the historical forest data from each quadrangle. So the next step was to find an extraction method so I can utilize the data in these maps and digitalize them. The first and most obvious method would be just tracing them, and you can see in this figure map B here, I started to do some tracing. This just one corner here took me about 12-15 minutes. And this is probably like 1/10 of a single map. And remember, there's 20 of them. So tracing wasn't what I wanted to do all summer long, and I don't think I would have learned much. So I looked for some other methods. The next method I looked at was pixel extraction, and that's here in map C. And basically you put in a set of values into GIS and it will extract pixels of that value, and they correspond with a color. So if you put in a range of pixels, it will extract those. And you can see it does a pretty good job of extracting the forest areas, but you can sometimes extract roads, some text. So it's not the best method. So I kept searching and found a classification-based approach. And this approach ended up being pretty good. I'll show it on the next slide as well. This breaks down the approach a little bit. So GIS has an easy to follow framework called Classification Wizard, which basically just runs you through all the available classification tools. So the first tool, segmentation. This is the original image A, and this is B after segmentation. And basically what it does is it groups similar pixels together based on color or shape. And you can see in map B that it successfully outlines a lot of these forest boundaries. So in the next step, the user actually has to teach the software what should be classified as each category. So my main goal was to teach the computer how to categorize forest and non-forest. So you can see that in the forest areas, I circled some green so it could understand that that area was forest. Then you run the classify tool and gives you an output of those two different values that I was teaching. And it does a pretty good job. You can see there's some problems, there's some grid lines that come through, and that allows you, and the last step allows you to fix that. It's called reclassifier. And it gives the user the opportunity to go back and, reclassify any misclassified areas. So it's similar to tracing, but it saves a lot of time. And then, so this is my final, product of all those maps together after they've been classified. This area down here is part of Harriman State Park. And this area up here is part of the Schwangdunks. So those areas, I thought there would be a lot of mature forest because those are heavily mountainous regions. And then you can tell in the middle of Orange County, this is a lot of the farmland and most of that deforested area. So there's less mature forests. So after that, I had to find a way to compare today's data with the data I collected. So I found some LiDAR data out from the Pennsylvania Geospatial Data Clearinghouse. And LiDAR data is light detection and ranging. It's a remote sensing method that uses light in the form of pulse laser to measure distances. So I used this data to compare with the other data, and I created my final map using a tool called CutFill. It's able to compare the two data sets. And you can see areas of dark green are mature forests, and then brown areas are deforested areas. This piece of land was deforested for farming at one point between the 1940s and now, and young forest areas are green. And I think there was about a 28,000 acre decrease in mature forest during that time. So that was interesting to hear. So my next thing I want to do is confirm some results. Actually, later today, I'm going out into the field to a couple of areas that are mature forests and doing some tree coring to verify results. But I did go out without a tree core just to look at some areas. You can see this is a snippet of my final map. This area here, the brown area is deforested. And this is actually now an elementary school that was developed, I think in the 1990s. So that confirms some of my results. And then this area up here, some mature forest. I went there and it was a forest with some swamp, which means that this area was likely never really developed because of that. So that was good to know. Then the last thing one of my main goals was to implement my data set into the conservation plan of the land trust I interned at this summer. So currently mature forests are not factored into the conservation opportunities format that's here. They basically put in a bunch of different maps with different parameters into one final overlay. So you can see things like land cover, elevation, be taken into account to calculate the opportunity score of conservation. So basically I want to put my map into that and see if there's any major changes, because it's important that we prioritize what to conserve, because you can't really conserve everything. So you may be wondering, what does this have to do with marine conservation? Classification approach can be used in really any setting, marine and coastal. The premise of my research can be applied to really any ecosystem with recorded vegetation data, as long as the area has available historical maps with forests and some sort of current forest or LiDAR data available. The method is repeatable. So for example, this map here, you can see I could actually look at the changes in the forest around Flax Pond, but it doesn't really have to be about forests either. Classification tools could be used to map out changes in high tide, for example, or analyze, the changing coastlines. So it can be used in really anything with the variable as time. So there was some limitations for my project. You can see on this map here, there's some areas of high density contour lines, and the approach that I use, sometimes it would misclassify an area like this as its own entity. So because it's so embedded into each other. Grid lines is also a problem. There's a bunch of grid lines here, but it does a pretty good job getting around that. And then there's a lot of text on this map in particular. So it could also misclassify some areas because of text. So really any overlying features makes it harder for it to extract that data. And as I mentioned earlier, these maps were created under the direction of the US Army to familiarize ourselves with domestic land in around the 1940s. I was unable to find sufficient information to know like the criteria of how they created these forest delineations. So maybe they only took areas for forests with areas over 100 acres. I would never know that. However, the project's goal is just to identify the location of these forests. So I still think that I'm able to do a pretty good job of that. And then there's some major importance with this. Identifying the age of forests influences, the value of protection and why we should protect those forests. The age of forests is difficult to measure without significant time, resources, and labor. However, GIS software offers a cost-effective and scalable classification-based approach that's able to identify the age of multiple forests in a short amount of time. Additionally, GIS classification tools are effective research approaches in any conservation field with change over time as the variable. The direct application of these results will provide conservationists with the location of mature forests in their specified area. Here's works cited. Thanks.
So we have about 2.5 minutes for questions. Maria?
Hello. I don't know if you can hear me, so I'm gonna talk really loud. But I commend you for your trial and error process with the GIS methods. As a fellow GIS user, I think that that's the most challenging part of any project, not just getting it to work, but getting it to work efficiently. I'm really glad you didn't spend your summer tracing all of those
Yeah,
points.
me too.
And the second, my second point is, as you said, your project is very scalable, and you created a very important tool that could be very specific to maps created by those cartographers, during that time. Do you have any plans to scale up your project to other areas?
Not currently, but I'd be open to it. I had a lot of fun with this project, and I think there's a lot more you can do with it. So yeah.
Okay, maybe time for one more. Paul?
Nice talk. I'm just curious if you're familiar with the satellite remote sensing product. It's called NDVI from Modus. It does the whole globe every 16 days at 250-meter resolution. Maybe that's too coarse, but have you looked at the satellite remote sensing data? And do you think it would be useful?
I haven't looked at that. I just use the LiDAR data just because I assume that that's the most accurate data I could find on existing forest data. But I haven't looked into that, no.
LiDAR would be much finer resolution, but Modus is global. It's updated, as I said, 16 days. You might have a look at it.
Awesome, thanks.
Sure.
Corinne?
Being relatively clueless about GIS, I was wondering if this same type of procedures and sort of programming and classifying could be applied to photographs, like drone photos and things like that.
Yeah, it's all of those images of those maps were all originally just a piece of paper. It's a photo that's uploaded to the computer. So you can then use the classification approach to extract any data that you want. The best way is there's differences in color, but yeah, you could do that with an image, yes.
Perfect timing. Okay, thank you.
Thanks.
Joseph Murphy is here and will be presenting next.
So we told him sitting down. Hello and welcome everyone. Good morning. This is the presentation for my internship as an assistant to Professor Finn's research into coastal reserve.
I just let me go rid of some of the extra.
Oh.
Oops. Ah.
Oops.
Sorry.
This is the foam logo right there. It's the red, green, yellow.
Let's try and get rid of this. Well, we'll leave it there.
Good morning again. This is the presentation as for my internship as an assistant to Professor Finn's research into coastal resilience and sustainability. Although the work here was done by me, the research design and structure was done by Professor Finn. I'm glad to have been a good help this summer and in a sense give back to the greater community here at SOMAS by being a direct help to the research of one of our professors. So before we begin, I'd like to scroll over a little bit of who I am and just talk about myself for a little bit, not to be too egocentric or anything. So my internship experience was chosen because I already had a decent amount of experience in the field of environmental planning and coastal policy when I interned at the Suffolk County Council of Environmental Quality for the summers of 2017, 18, and 19. This was also my an internship assignment for my undergrad when I was an undergrad here for environmental studies. I wanted to expand on this interest as well as my expertise with working with Professor Finn for coastal resilience and sustainability research. The prior internship those past summers gave me honed my skills hone my skills better in Microsoft Excel and Google Sheets, which were required for this internship this summer. And thanks to the previous internship, my familiarity with SECRA, which you can see is outlined in the slide, State Environmental Quality Review Act, came useful when reading the multitude of municipal documents relating to the environment for my work this summer, at least for the ones for New York. Other projects I've done prior to the summer session were assisting the development of the Blue Waste Trail project, which is defined on that bottom bullet there on the slide. And with that, I'm gonna go a little bit more. So in my time working with Professor Finn, I was addressed with the following questions, and he made it clear that these are the questions I should have in mind when assisting him with his research, which is, what is the state of coastal sustainability and resilience planning in the Northeast US? What are local governments doing to address climate change mitigation and climate change adaptation and risk mitigation? And what are the best practices and challenges or barriers regarding these efforts? So why is this important? Well, I'm sure many of you may know that coastal environments are the most extensively used regions of the world when it comes to supporting urban populations, industries, and other types of activities. Even in the United States, the most populated cities are in some way adjacent to the coast, such as New York City, Los Angeles, even Chicago is on the coast of Lake Michigan. Extensive use often comes at the expense of the natural environment. And so the main challenge we're trying to look at here is to see if we can find a balance between the needs of society, civilization, as well as the preservation of coastal ecosystems for the future. Essentially the basic fundamental tenets of what it means to be sustainable. The concept of coastal resilience has been brought more to the forefront as populations of coastal urban settings continue to increase, as well as us becoming more cognizant to the effects of climate change in recent years. Also, the emergence of storm events from the likes of Katrina and Sandy have led to many municipalities becoming more proactive in bolstering the defense of the coast in order to protect infrastructure as well as many natural components. So to give an example, I'd like to look at New York City, which is an area that has been met with an accelerated rise in sea level to around an inch and a half rise per decade. It is in the top 10 in terms of assets exposed to coastal flooding in the United States, second only to Miami. So for this area, Superstorm Sandy represented the first major disaster where recovery planning has been used overtly as a tool, not just for rebuilding and repair, but for enhancing future resilience to the Big Apple. So new slide. Here we go. This might be a little bit long. Let me see if I can manually. As good as it's going to get. All right. So in terms of research tasks and assignments, the assignment that I worked on the most, that I spent the most time on, was the coastal Northeast Communities and Plan 2020. My time with this task was entirely dedicated to researching and locating municipal and planning documents for over 500 different coastal communities along the Atlantic and New England coast. I was to find the resilience and sustainability plans of each community, the comprehensive plan, and any climate action or adaptation plans I could find from my research, as well as place them in an organized Microsoft Excel document. In order to find coastal communities and municipalities, I used mapping software such as Google Earth to locate them. The states ranged from New England to the northern Atlantic states, such as New York and New Jersey. Some documents, when searching for the documents, some of them were as easy as a Google search, and a good number of them I had to do quite a bit of digging to find, usually by looking through the e-codes or through online archives of some sort. Finally, the last project was to analyze the varied documents I collected previously and code them appropriately based on certain criteria. In my analysis, certain questions were asked, such as, oh, actually, well, I didn't go with the right slide. I actually, my bad. So here this is just a good visualization of what a finding the municipal plans of a city would look like. This is Boston, as you can see. This is just what the average Excel document would have looked like for a city. And then, so in my coding, I wanted to analyze and research what are we looking for in these plans? I want to see if I can address certain questions, such as, does this plan address stopping pollutant runoff? Or are there any contingency plans in case of major disasters, or are there density bonuses to develop in non-coastal areas, and so on. So here's the preliminary results. The number of coastal communities I found in my first assignment was around 575. Keep in mind, I was assigned to find the coastal and riverside communities of New England, New York, and New Jersey. 65% of them had an identifiable comprehensive plan, and over 80% had a resilience plan, and over 80 had a sustainability plan. But one thing I found a bit concerning is that less than a quarter of the communities had a written or quantifiable climate adaptation plan, and less than a quarter of them had a climate action plan. The next step is to look carefully at all the other plans and better understand the content and what issues are currently being addressed. So this is a table summed up, which I scored the following for questions of what we're looking for. You'll see the questions that they were shortly, but these are just the scores of 25 random sampled communities, and I was evaluating them based on their comprehensive and master plans. I was able to score them in the table above, and before going into this, originally I had hypothesized that the communities with the highest populations would score the highest in terms of plan evaluation. And while the lowest populations generally were on the lower end of the spectrum, well, except Beals, which is a bit of an outlier here at sitting at barely below 500. Boston seemed to be a great outlier in terms of population. Even then, I suspected that it would be the top, but it's top 5, which not quite. Not only that, but the type of municipality didn't seem to influence the overall score either. So this table and chart show that there does not seem to be too much of a preliminary relationship between population size and plan score for our initial sample. But it's possible that the sample is also too small to pick up on these relationships. So this is something we would like to look at moving forward. You can note here that the smallest community, which is Beals, Maine, right here, was the 10th highest scoring plan, while the largest city other than Boston, which is Schenectady, ranked 20th, as you can see here, which, let's see, where are you? Largest city, oh, right, here you are. Second largest city, and it only scored 19 out of 50. So these are the questions that we answered for strategies. You can see here, pollutant runoff, coastal development controls, stormwater flooding strategies, floodplain retreat, and so on and so forth. So after analyzing the results of the plan evaluation, we come to the statistical conclusion to many of the strategies we were looking for when reading through the various plans. As you can see, the strategies concerning pollutant runoff, planning participation, and stormwater flooding strategies dominated in terms of detailed plans present. Meanwhile, the strategies concerning development moratoria, environmental justice focus, and the removal of gray infrastructure were the least present in all the municipality plans analyzed. One of the statistics that I thought was a great note and a bit of a sigh of relief was that there's exactly 0 mitigation plans that aren't present. So mitigation requirements is particularly, they're present on every single plan that we looked over, which of course is a good thing. Not only that, but pollutant runoff and stormwater flooding strategies are also the most common, which generally are the most fundamental strategies for coastal resilience and sustainability. So I saw that as a net positive to have those on the higher end of the scoring, because it's just the bare fundamentals in that case. And so to explore the communities, I've looked at one high score and one low score, which is town of Southold and town of Northport, which not the Northport here, but Northport, Maine. So here I've chosen 2 communities that are on the opposing ends of the scoring spectrum. And right off the bat, one thing I noticed is the dates of the plans, which I say might be a factor here. And be reasonable to assume that this would have an impact on the scoring system as well. Both of the communities are at risk of flooding according to their municipal documents. However, the former has addressed it far more proactively in detail than their regional planning. For future work, it's best for us to try and understand. Okay, just one more slide. We can hypothesize that population, geography, and a plethora of other factors that can serve as explanations, but for now, it should be considered as the next step in our research. So implications and next steps. We finished our plan collection. I intend on working with them as well after the fact. So our next step is to finish the plan collection of other jurisdictions. Maybe we can start looking at counties, regional planning bodies instead of just the cities, towns, and villages. Maybe develop sampling and plan coding strategy. And the main question I got from this takeaway was, what is the reason some communities score higher than others? And that should be a key factor in our analysis down the line. These are just the sources, and this concludes my presentation. Thank you all.
We have some time.
Yeah, any questions? Yes.
Can you tell us more about your scoring? Do you have a system? How do you score?
Yes. Accidentally took it down, but the scoring was based on a set of questions that we would look at when it comes to scoring the plans here. These strategies in this box here, these are the questions we look for. What are the mitigation requirements? Is one of them. Is there an environmental justice focus? Are there coastal economic development programs? Is there any plans for the state to purchase or acquire coastal land? Are there any ongoing risk analysis for properties and so on? So you see the questions are just this general big box right here. That's labeled strategies.
So you just have to score the quality of the plan?
Yes, based on what was present and what wasn't.
Okay, one more quick question.
Oh, yes.
Who came up with the scoring criteria?
That was the work of my advisor, Professor Finn. You see him in the back. There he is.
So that's drawn from the growing literature on the topic. There's 25. There's basically it's not for the category of that's a very short description of the longer description. 0, it's not present at all. It's sort of mentioned in passing, so there's some acknowledgement of municipalities thinking about it, but not really anything detailed that it's a one. It's a standard plan evaluation.
No, just to clarify, you're looking at whether or not certain strategies or topics are covered, not the quality of it, right? Is there a distinction?
Covered, and then a very coarse measure of detail. So present versus like a detailed action or strategy attached to it, as opposed to second.
Okay.
All right. Once again, thank you, everyone.
Okay, our next speaker is Nicole Dergosits.
And it is more longevity in deep sea and non-surface polar environments. The global popularity of plastic has resulted in worldwide pollution on land and in sea, affecting both humans and animals. Plastic litter is found in terrestrial environments, in the open ocean, and on shorelines of the most isolated islands. Yearly plastic production has already matched the combined weight of the human population. In 2015, approximately 6.3 billion metric tons of plastic waste was reported, and 9% of the total waste was recycled, 12% was destroyed, and 79% ended up in landfills or in nature. Plastic waste is entering the ocean at a rate of approximately 11 million metric tons per year, and experts have predicted that there will be more plastic than fish in the ocean by 2050. An examination of 340 original publications revealed that at least 690 different species have been negatively affected by marine debris, which is 92% plastic. Animals can accidentally ingest plastic particles and suffocate from entanglement in plastic debris when they get stuck in products such as plastic bags, packaging, and nets. More than 640,000 tons of ghost gear from the fishing industry end up in the ocean every year, and most of the gear is plastic. Balloons and plastic bags are especially harmful to marine life, but ghost gear is an even bigger threat. Some kinds of marine organisms are especially vulnerable to plastic waste, such as turtles, marine mammals, suspension feeders, and deposit feeders. Seabirds are also highly likely to consume plastic, and their stomach contents are used to reveal the health of marine ecosystems by the Oslo and Paris Convention in the North Sea. 60% of full-Mars seabirds have at least 0.1 grams of plastic in their stomachs, which is considered 6 times more than the target threshold as of 2017. In response to the plastic crisis, from the years 2000 to 2019, there have been at least 28 international policies set up to reduce plastic pollution. However, these efforts have not been enough to slow down the rate of plastic pollution. The execution of law is split across national and local jurisdictions, which results in regulatory disparities in world environmental governance. Current international rules, state policies, non-state rules, and consumer behaviors are not adequate enough to accomplish an impactful reduction of plastic use we see on a global scale. This chart shows the share of global plastic waste emitted to the ocean. Asian countries have emitted 80.99% of plastic waste to the ocean. China manufactures the most plastic in the world. However, the US is responsible for bringing an estimated 0.51 to 1.5 million metric tons of plastic waste to the coastal environment per year, and has a long history of shipping plastic waste overseas to China and developing nations that are not able to properly manage the waste. During recent years, there have been effective bans on plastic bags, straws, etc. in several locations. However, the COVID-19 pandemic had created a setback. Some governments and businesses didn't keep their commitments to tackle plastic pollution. Some companies even banned reusables completely as a safety precaution. Additionally, improperly discarded personal protective equipment has contributed to the litter found on land and in sea. For instance, tens of disposable masks were found in a 100-meter stretch in Sokos Islands Beach, Hong Kong, during an environmental survey that was conducted by the NGO Ocean Asia. In 2015, Indonesia was estimated to be the second largest country that contributed the most to plastic pollution in the marine environment following China. The pollution has been unmanageable, with 14% of the solid waste components in Indonesia being plastic, and the solid waste management infrastructure and services are still insufficient. The lack of resources and management in certain parts of the world significantly increases global plastic pollution. The Surfrider Foundation has been working towards combating plastic pollution since 1984. Surfrider is a nonprofit grassroots organization with a growing volunteer activist network. There are 80 chapters across the country with over 50,000 volunteers, 50,000 members, 100 student clubs, and over 1 million supporters. Their mission states, the Surfrider Foundation is dedicated to the protection and enjoyment of the world's oceans, waves, and beaches for all people through a powerful activist network. The Surfrider Foundation actively works on campaigns for reducing plastic pollution, ocean protection, beach access, coastal preservation, and clean water. Surfrider's model allows them to make changes on multiple levels. By having a network of chapters across the United States, local level changes are possible with community involvement and collaboration with local cities and governments. Every coastal region in the country has Surfrider experts to bring chapters together when problems are too big for a single chapter to tackle. The foundation also has a team of legal, policy, and environmental professionals who work with federal agencies to create change at a national level. When the team wins a campaign, it is considered a victory. Surfrider defines a victory as a decision by a governing body that protects the coastal and ocean environment, improves coastal access, or both. The foundation has had over 700 victories to date. The foundation aims to reduce the impact of plastics in the ocean, to protect our ocean from threats to the ecosystem, partner with decision makers to allow full and fair beach access for everyone, tackle issues that endanger beaches and natural shorelines, and maintain the health and sustainability of the ocean. Surfrider promotes solutions such as reusable alternatives, joining beach cleanups, and eating at ocean-friendly restaurants. Surfrider regularly hosts film screenings of educational and thought-provoking documentaries they think would inspire people to take action. Surfrider also asks the directors of the films to join the film screening for a Q&A with the viewers to start productive discussions about how we can take action. Rachel Casilla, Surfrider's plastic pollution manager, is currently working on a summer film series and asked me to help her with the planning of this project. The goal of this film series is to distill messages to the network and help frame the issue of plastic in a more holistic and uplifting way. Voice Above Water was the first film I watched to prepare for the film series. It has a universal message, but it brings attention specifically to the plastic crisis of Indonesia. It is a short yet very powerful documentary that reveals an unsung hero fighting plastic pollution. A 90-year-old fisherman named Wayan Neo has devoted the past 21 years of his life to collecting plastic from the ocean using his small fishing boat and net. He hopes that someday he'll be able to catch fish again instead of plastic. My capstone project fits in with the larger question of what can we do to improve waste management infrastructures and services in Indonesia? We should not place all the responsibility on the people of Indonesia, since tourism is a large contributor to the pollution there, according to film director Dana Frankoff. We need to encourage the tourists to use less single-use plastics by providing convenient alternatives. Dana agreed to let Surfrider host free film screenings of her documentary for Surfrider chapters and clubs this summer, and I recently did an interview with her to get more people interested in watching Voice Above Water and to raise more awareness about plastic pollution. You can find the interview in my blog post on Surfrider's website. World Refill Day is an international public awareness campaign supported by the European NGOs Coalition Movement, #WeChooseReuse, which Surfrider is involved in as well. The goal is to normalize refilling and reusing instead of using single-use plastics and to reduce plastic pollution globally. As a way to make it easier for consumers to be more sustainable, a free app called Refill was developed to connect people to a global network of places where they can reduce, reuse, and refill. People can now find out where water refill stations and public water fountains are located, and find coffee shops and restaurants that offer discounts and rewards for bringing their own reusable cups and containers. The app developers reported that 270,000 refill stations have been added worldwide following the success of World Refill Day. The people behind the campaign are effective at encouraging both individuals and businesses to change their behaviors by providing convenient methods to reduce plastic usage. I did a post-campaign analysis of World Refill Day on social media, identified every Surfrider chapter that participated in social media posts about World Refill Day and which platforms they used. For my analysis, 13 chapters posted on Instagram, 14 chapters posted on Facebook, and 2 chapters posted on Twitter about World Refill Day. I also quantified how much engagement each Surfrider post got on each of the 3 platforms. After completing the analysis, Rachel also asked me if I noticed the type of messaging being used in my review, and if there was a common thread or messaging that seemed to get more engagement. I found that an Instagram post by Surfrider San Diego got the most comments out of all the Surfrider posts about World Refill Day on Instagram, Facebook, and Twitter. People commented about which places have bulk bins, and another person commented about a coffee shop that uses reusable cups. It will be effective if people know which coffee shops, restaurants, and stores are eco-friendly and allow reusables. The people who commented on the post are enthusiastic about spreading the word. My post-campaign analysis for World Refill Day will be used as a model for future campaign and social media analysis for Surfrider, according to Surfrider's legal director. Humans are routinely exposed to plastics in personal care products, household items, and clothing, but it is reported that humans consume between 39,000 to 52,000 microplastic particles every year from food and drinks alone. Compounds such as bisphenol, phthalates, non-persistent pesticides, perfluoroalkyl chemicals, and perchlorate are considered risks to human health and are permitted to come in contact with food, primarily through diverse forms of plastic packaging. We need to further our understanding of the bioavailability of chemical contaminants found in plastics to be able to assess their risks to human consumers. The Surfrider Foundation has an ocean-friendly restaurants program, which recognizes restaurants that are making sustainable changes. The Surfrider Foundation states that plastic contains toxic chemicals that can transfer from cutlery, containers, cups, and straws directly into our food. The restaurants on Surfrider's list are dedicated to eliminating single-use plastic products in their businesses. The minimum 7 requirements to be listed as an ocean-friendly restaurant are only using reusable foodware for on-site dining, providing plastic straws are provided only upon request, no expanded polystyrene use, aka styrofoam, no plastic bags are used for takeout or to-go orders, single-use utensils, straws, condiments, and other accessory items are provided only upon request, beverages are not sold in plastic bottles, and proper recycling practices are followed. The Surfriders Foundation website currently lists 672 ocean-friendly restaurants nationwide. From what I have learned from doing my capstone project, it is clear that recycling is not enough of a solution. We need to work harder towards reducing and reusing plastics. Individuals should decrease consumption and limit their plastic waste, and industries should be accountable for the waste they create and design products more sustainably. It is estimated that 50% of plastic packaging could be recycled if packaging designs are recreated. Also, if industries use clear or light colors in their products, it will improve the quality of recycled plastics. Furthermore, governments should spread public awareness about plastic pollution, enforce restrictions, and improve plastic waste management services. Increasing behavioral benefits and decreasing behavioral costs of reducing plastic waste for decision makers would be a necessary part of the solution. It is vital to improve our understanding of behavioral incentives and to incorporate that knowledge in policy making. The Surfrider Foundation is a prominent national resource for combating plastic pollution locally and nationally. Surfrider is not trying to convince people to completely remove plastic from their lives, since it is almost impossible to avoid plastic products. Instead, Surfrider is trying to encourage people to be mindful of their choices that impact the environment and inspire people to change their lifestyles. I have been interested in learning from people who are successfully reducing their environmental footprint, and I encourage you to watch 2 other documentaries I watched as part of my project, Living in the Future's Past and Living the Change. Those documentaries explain the severity of the global problems we are facing, but they are also inspiring and motivating. Living the Change shows people who are truly being the change they wish to see in the world. They are living alternative lifestyles that are more minimalistic by implementing forest gardens, composting toilets, community-supported agriculture, and time banking. Living in the Future's Past discusses how all of us are connected to our environment and to each other. I used what I learned from these films for the mindfulness minimalism aspect of my project. In conclusion, plastic pollution is a widespread and overwhelming issue to tackle, but change is possible if we improve our mindsets and our behaviors for the well-being of our planet and future generations.
Thank you, Nicole.
That was actually 15 minutes, but maybe we could have one question.
Nicole, are there any questions?
Say that again?
I said, are there any questions here for Nicole? No, okay. Well, thank you very much, both of you, for nice presentation and very clear presentation of the presentation. Okay. So
Okay, next we have Steve Silverman. You've already seen him today in the video that was shown by Amanda of the trawling on the Shinnecock Bay Research Program. But his project is different.
All right. Hey everyone, how are you doing? My name is Steve. So my project is titled The Role of Aquariums in Marine Conservation, and my SOMAS advisor was Ellen Pikitch for the summer. And then I worked at the Long Island Aquarium out in Riverhead, and my advisor there was Colette Ferraro. She is the head of the education department there. All right, so I just want to give you guys a little bit of a background to my life and why I chose to work at the aquarium and focus on aquariums and conservation as my project for the summer. So I went to Binghamton as an undergrad, and during my time there, I got the opportunity to work in Woods Hole at the Marine Biological Laboratory. And besides learning about a lot of cool science stuff there, I really learned how important public outreach and public education and science communication are. So a lot of the things that come out of the science world really only make their way to other scientists, and a lot of scientists just write their results and stuff like that in kind of like diction that other scientists interpret and use all the time. But a lot of people in the normal world that aren't in the science world, you know, don't really understand a lot of this. So public education and science communication is super important, especially when things like the IPCC AR6 report come out that just show that, you know, now is the most important time to take action, and public education is at the forefront of that action. So with public education and science communication, there are a couple different forms of it, including public outreach. So like a group going to a beach, for example, and describing what they do. For like example, Atlantic Marine Conservation Society. They'll go to some beaches and just talk publicly about like what they do. And so the next thing is citizen science organizations. So that's pretty much just taking citizens out in the world and using them in science. So some organizations will actually take them in and let them do some work. And then there's other forms like iSeek or FishBrain, which are apps where you could actually report fish sightings or animal sightings, insect reportings. And then scientists will use that data to get a general idea of like the range of species, stuff like that. And there's also some school programs, but finally I want to focus on aquariums. So during my time here at SOMAS, I did a lot of research into all of these forms of public outreach and education. And there are a decent amount of research into all the categories except for really aquariums. There's not too much research on how effective aquariums are at science communication. So teaching all of their guests all about marine conservation, the species that they have there, and the habitats that they have there. So education as a means of conservation. So Reed et al. 2021 was an article that pretty much came to the conclusion that many people consider education to be one of the most important and effective drivers of large-scale social change. And at this point in marine conservation and just environmental conservation in general, really what we need to see is this large-scale social change. A lot of the small changes are not going to be as effective at this point. And so an increase in education, environmental education, leads to increased knowledge on the issues and solutions at hand. So the people that are learning and being educated are now going to have an increased knowledge about all of the issues that marine conservation and conservation in general face. And then some of those solutions that we could use on a small scale or large scale. Additionally, it leads to a greater awareness and sensitivity of these issues. So for example, things that you do in your life and how they relate to large scale issues like recycling and pollution. And then finally, so it creates a change in attitude and a drive to help. So a lot of the time, when you're educated on a topic, you know, you'll kind of get a connection to that topic and you'll be more inclined to help. For example, conservation. If you learn about a species, let's say that is local here, it's like a harp seal or something like that, or a harbor seal, then you'll be more inclined to actually take action and help once you do learn about it. All right. So the next slide is titled "The State of Aquariums and Their Role in Conservation." So some of the benefits of aquariums are they provide access to a lot of the species and habitats that landlocked people or people who have an inability to access transportation or travel would see. So a lot of people, they do live in landlocked areas and there's not an opportunity for them to readily go over to the ocean or some body of water and, you know, just interact with it, see what's there, kind of form a connection between that. So that removes that that like roadblock essentially. So the next one is allows guests to form connections with nature. So seeing all this stuff in person allows you to form a better connection than just seeing it, for example, online or in a picture or just hearing about it. So actually being able to see the animals and the habitats that are being talked about in conservation allows you to form a better connection. And then finally, aquariums take part in these crucial conservation and breeding programs. So a lot of people overlook this aspect of aquariums. So for example, at the Long Island Aquarium, one of our breeding and conservation programs that we focus on a lot is with our African black-footed penguins. So they became critically endangered in 2010 due to overfishing and pollution. And so our facility is one of many facilities in the country that focuses on breeding these African black-footed penguins so that their population is not going to decline. So one of the, like I said, there's been really a lack of research on how effective aquariums are at science communication and public education. However, there was one comprehensive paper that came out in 2007 entitled Why Zoos and Aquariums Matter, Assessing the Impact of a Visit to a Zoo or Aquarium. And this is really one of the only comprehensive studies that I found, or even studies at all that I found. So this study pretty much looked at 12 AZA, or the Association of Zoos and Aquariums, accredited facilities. So there are more than 500 zoos and aquariums in the United States, but only 10% of those are AZA accredited. So it's really a small group that they're looking at. And over the course of their study, they had 5,500 participants that actually completed their survey. So the results that they got from their survey was that, so they focused on two things in their hypothesis. So first, that a visit effect on their guest knowledge, so how much the guests learned. And then secondly, how a visit affected their guest habits. So if they're gonna change some of their habits and behaviors in their daily life to reflect a more environmentally friendly lifestyle and lower carbon footprint. So the results found that there was an overall positive effect on both of these. However, they had no quantitative measurement of this. They only asked if their participants believed they had a greater knowledge or believed that they had a greater inclination to change their behaviors. They didn't actually have a quantitative measurement of that, which is where my study comes into play. Okay, so first I just want to go over my role at the aquarium. So I was part of the education department. So through the education department, I would answer questions. So I'd be placed at various stations throughout the aquarium and just answer questions that all of our guests had about the habitats or species. I would also give lectures. There's several habitats that we give lectures at, for example, the Stingray Bay, the shark habitat, the lagoon, African black-footed penguins, our North American river otters. So during those lectures, I would pretty much just talk about the species and the habitat relating to their biology and physiology, some cool adaptations that they have, and then finally, some species-specific conservation facts. Additionally, I would teach classes. So for example, one of the most recent classes I taught was for the Cove Library, and that was a class on horseshoe crabs. And then finally, I would take part in some animal husbandry. So I was in charge of a lot of the reptiles that we have there. So part of the education that we do there is taking out a lot of the animals so that the guests can actually interact with them physically and gain that connection through them. And then they have a greater connection or maybe an inclination to care about those animals. So like I said, the one comprehensive study that there was out there had no quantitative measure of their results. So I really wanted to kind of quantify my results through the Long Island Aquarium. So to do that, I created a two-part survey. The data collection period was from June 14th to August 9th this summer. So part one of the survey asked some questions about guest background, like, have you been to the aquarium before? Have you been to any aquarium before? How far away you live, stuff like that. And then also, more importantly, it asked 9 specific questions about these species and habitats that we have at the aquarium, specifically the ones that are lectured on. And then part 2 asked questions about guest behavior, whether or not they took part in some activities before the aquarium and then after. And I'll go over that in a minute when I go over the actual survey. So these surveys were distributed in 2 ways. First way was virtual, the second way was in person. So the virtual one was so that when I wasn't at the aquarium, people still had access to the survey. So I would put up I put up signs at each of the exits that had a QR code. I'll give an example of the sign and survey in a second. So pretty much when people left the aquarium, they had access to this sign that had the QR code so that they could scan it and take the survey when I wasn't there. But when I was there, I would go around in person and talk to all of our guests, give them a little spiel on my project and why it was important, how they can contribute to my research through taking the survey. And then I would hand them a flyer and show them how to scan the QR code. So that was the second way. So there were 524 flyers handed out in total. So I talked to 524 people. However, only 130 completed the survey. So that was kind of lower than I expected and was definitely lower than the amount of people exposed to the survey. So this is an example of the sign that was put up at each of the exits and also the survey that I handed out. So a couple of important things that I just want to touch upon that were touched upon in previous papers that hand out surveys like this. So you want the QR code for the survey to be like the main thing in the middle so that people see it. And then also having the, on the left where it says only 5 minutes, it shows that, you know, it's not going to take too much of your time. So guests will be a little more incentivized to take it. And also with incentives on the right-hand side, you can see it says a chance for, if you fill it out, you get a chance to win free entrance through a raffle. So that's another incentivization for the guests to fill it out. And then finally, at the bottom, it just says survey for master's capstones at Stony Brook. So my boss at the aquarium, she said people in the past had done this. And when they relate things back to the aquarium, people are actually less inclined to fill it out than it was for somebody's personal information or personal research, which is why I heard that. Okay, so these are just an example of what the survey questions look like on the Google survey. So on the left is from part 1. So one example of a species specific question that I would ask. This is the top one. It says the use of DDT had an extremely harmful effect on the eggs of which species of seabird on Long Island in the 1900s. So ideally, if one of the guests came to the lagoon habitat and read the signs or attended the lecture, they should be able to know the answer to that question. And that is the way all of our questions were designed. They were pulled from the signs or the lecture material so that the guests would have the best opportunity to answer them correctly. And also there's some other questions like, did you visit the Marine Center today? Or did you talk to or ask any questions to the education staff? And then on the right side is the second part of the survey. And like I said, I just asked about behaviors. So the first question, for example, is have you ever cleaned up a beach or participated in a beach cleanup? And then the second one is after visiting the Long Island Aquarium today, are you more likely to clean up or participate in a beach cleanup? Okay, so my hypothesis first, I was visiting the aquarium for the first time, reading signs, attending lectures, and talking to the education department would all lead to higher scores on the survey. So reading the signs, attending the lectures, and talking to the educational department all will provide the guests with access to the information that the questions are about. And also, I thought that visiting the aquarium for the first time would lead people to be more likely to actually pay attention to the lectures and go to the lectures and read all the signs, rather than someone who comes there all the time and has really like seen the lectures before and has seen all the signs, stuff like that. So my hypothesis for part 2 was that visiting the aquarium will make guests who don't already take part in all 4 of the activities that I selected, which were recycling, using organic fertilizers, eating sustainable seafood, and participating in beach cleanups. And the reason I chose all 4 of those activities is because they are highlighted in our lectures and signs of the habitats. Okay, so the results from the lectures. The top row is going to be the lecture attendance, which was surprisingly really low. So the lowest was the shark in the lagoon habitats. Those lectures, only 6.2% of the 130 guests that were surveyed went to those lectures. And the highest was our sea lion show at 53.1%. So looking at the next couple of rows, you can see broken into 4 different categories. Those that attended and got the question correct, those that attended and got the question wrong, those that didn't attend and still got the question right, and then those that didn't attend and got the question wrong. So I was going to expect, based on my hypothesis, that this percentage, so the percentage of people that got it correct, would be higher than the percentage of people who went and got it correct. However, in only 3 of the questions, that was the case. So for the Ray 1 and 2 and Shark question 2, those that attended the lecture got the question correct at a higher percentage than those that didn't attend the lecture. And that was kind of surprising. And then, so the t-test results, I just did 3 t-tests on 3 different groups. So those that attended the aquarium previously, or were attending aquarium for the first time, those that read the signs and those that didn't, and then those that talked to the education department and didn't. And the only significant result was from those that talked to the education department. Their average score was about 6 points higher. And then finally, the habitat, or the habit or behavior results. So actually, the majority of people for recycling using organic fertilizer and beach cleanups already have participated in all of these activities beforehand and are still likely to participate after. However, the one that didn't have the majority of people already participating in it was sustainable seafood. And the majority in that section selected that they didn't do it before, but they are likely to do it afterwards. So then, just in conclusion, there was only a significant difference between talking to the education department and not, which is kind of cool because I'm in the education department, so it's nice to see that my job has an impact on people. Like I said, there was no significant difference for those who attended the aquarium for the first time or reading signs. And then attending the lectures only increased the percentage in 3. It was kind of inconclusive. So more research needs to be done in these small private aquariums. They are the majority of facilities in our country. There's really no research on them. And in the future, if someone's going to work on this project, they might want to consider a different way of collecting data and also possibly reaching out to these guests in the future to see if their answers were the same as the day that they came to the aquarium.
That's fine. We ran out of time, and what I'm going to say is, like, during the lunch after this, feel free to ask Steve questions. It is kind of perplexing, the results that you got. So I'm thinking about why would this be? Next speaker, Sydney Randall.
Everybody, my name is Sydney, and my capstone project was called Social Media in the Conservation Field, a Science Communication Case Study with the Red Sea Reef Foundation. My advisor was Dr. Quinn Clinthouse. So the Red Sea Reef Foundation is a new nonprofit focused on supporting groundbreaking research on the Red Sea's uniquely resilient corals and promoting regional and global scientific collaboration to advance coral science and conservation. It was founded in 2020 by Dr. Quinn Clinthouse. And basically, she reached out, she wanted to expand its reach, build a broader base of support, and really just let the world know about this reef. So I conducted a 3-month-long research-based social media campaign for the Red Sea Reef Foundation. I also created a list of recommendations through research and my results for other conservation and science nonprofits for their social media strategy. And this was a really great science communication challenge because the RSRF was so new. So there weren't any of our own funded projects to support or to promote. And also we were basically starting from zero with a couple of the platforms. So it was a really cool way to see that growth kind of starting from nothing and expanding. So I had 4 main goals for this campaign. One was to spread scientifically accurate information about the properties of the Red Sea Reef. 2 is to highlight different conservation challenges the Red Sea Reef is facing and promote policy solutions. 3 was to increase donations for the nonprofit. And 4 was to build and engage social media community on Twitter, Facebook, and Instagram. So as we all know, social media is huge today. A recent study in 2019 found that most adults cite the internet as their main source of news. 69% of all Americans use Facebook, 40% use Instagram, and 23% use Twitter. And around 3.78 billion people across the world use social media. So really, it's just going to continue to expand. So it's really crucial that organizations and nonprofits and scientists know how to effectively utilize this tool and reach a really wide audience. We're also entering sort of a new media landscape as social media continues to expand. And contrary to what we might think, public perception and trust in scientists has actually remained relatively high and stable over the past 10 years. So it's about 40% of adults in 2016 still feel a high level of trust. And when you compare that to only 7% of adults feel trust in Congress, that's pretty high. So it's actually kind of encouraging that people still have trust in the scientific community, and that number is actually still going up. And basically what we're seeing is that the deficit model, which is the long sort of way that people would communicate science by just giving people information and facts and then expecting them to become more scientifically literate and make the right decisions in conservation fields, that's really not working anymore. And we have to sort of transition to a more involved, engaged model, like the public engagement model, which is all focused about community capacity building, working with the people, and having a sort of back and forth. Or the lay expertise model, which breaks down the barriers between scientists and layperson, incorporates all of the knowledge that the audience has, brings that into what you're talking about. And again, it's more audience focused versus speaker focused. And social media is a great way to do that because it's all interactive. So why the Red Sea Reef? Red Sea Reef is a very special place. It stretches 4,000 kilometers along the Red Sea shoreline. It supports over 28 million people in terms of food and livelihoods. The economic value of the fishery is $230 million per year. Tourism goes up to $12 billion per year. It's a biodiversity hotspot. It supports a ton of endemic species. And also the unique adaptations of the Red Sea Reef produces different molecules and metabolites that could potentially not be found in other systems and could be a major source for medicines and other natural products. As we heard earlier from Amy, coral reefs across the globe are degrading rapidly. It's estimated that 70 to 90% of all coral reefs will be majorly degraded by 2050. And another really special thing about this reef is that it is so long and it stretches across this really intense latitude from 12.5 to 29.5 degrees. So it sort of creates a natural laboratory, this environmental gradient, where you can see how corals respond to different temperatures right there. In the Gulf of Aqaba, which I've circled there, is the northernmost point, and it hosts these incredibly heat-resistant corals. So they have a hypothesized incredibly high thermal resilience, which means that this reef ecosystem could be a potential marine refuge in the face of climate change and could hold the biological key to the survival of the world's remaining reefs. So basically, it really needs to be studied and protected, but there's a lot of different threats facing this reef, including urbanization, coastal development, polluted runoff, declining in water quality, overexploitation of fisheries, and then, enter the Red Sea Reef Foundation, who's working really hard to promote that research that's going on in that area, as well as get the Gulf of Aqaba declared a UN World Heritage Site. So when designing my campaign, I designed it for about 2 months, and then we conducted it for an actual 3 months over the winter. We went for 3 to 5 posts a week, and I decided on that number. We decided together for a couple of reasons. One, there is a lot of data out there about you don't want to overwhelm the audience. There's different numbers of how many times you should post for each platform. And also I wanted to make it a sustainable number so that the RSRF could continue being active on social media after I wasn't doing the campaign any longer. I looked at other organizations like Sea Legacy, Nature Conserves, highlighted some high traffic hashtags, found some common themes in my research and through those other organizations. And I used this tool that I learned from the All-in-All Center for Communicating Science, which is a great way to identify the audience that you wanted to reach, what goals you wanted to have, why might they care, what are the obstacles, etc. I also reached out to the Stony Brook social media team, just Mark Lang, asking if he would share our post, just to sort of spread that engagement even more. And in order to reach the audience that we wanted, which was, idealistic people, young people who are interested in learning more about this reef, potential donors, so people who would be willing to actually make donations to the page, as well as other organizations and other nonprofits, so we could build that sort of camaraderie and community among this group. I decided to use political, economic, cultural, and environmental frames, all the while trying to create sort of an emotional, values-based connection with all of these people. These are the common themes that I found, which I'll talk about more later in my final recommendations, but I then created a schedule. So I started this Coral Critter of the Week project campaign thing. So every Monday we would highlight a different creature that lived or fed or breeded in the Red Sea Reef, and we would kind of make it like a fun way for people to connect to the things that live there. And that was actually a really successful project. Other than that, I worked on different, call for donations, talked about the functions of coral reefs in general, talked about the threats facing corals, the threats facing the Red Sea Reef. We did a couple board profiles, and then stories of coral research in the field, not that we were funding, but that we would hope to fund in the future, things like that. So moving on to the results. So Facebook had the least amount of growth. I did 41 posts to Facebook. We saw a 43.2 increase in followers, 95 to 135. And I calculated engagement, which Nicole talked about in hers, but I calculated engagement, which is just a metric. You take the average number of interactions with each post, so like likes, shares, comments, etc. You divide it by the number of followers that it has at that time, and then you just find the percentage. So our average engagement for Facebook was 7.47. So that means about 7.5% of all of our followers over the 12 weeks engaged with our posts on average. So that's actually really high. I found this study that looked and found the industry average for nonprofit Facebook groups. The average is 0.13%. So we were really good in terms of engagement. Also found that our likes grew very slightly over time. There wasn't a huge increase of likes over time, and our shares actually decreased over time, again, very slightly. So those were kind of remained relatively stable throughout. So for Twitter, I did 50 tweets, a little more activity on Twitter. We actually had an 800 increase in followers from 15 to 135. Also had an increase in amount of likes and retweets on the post as time went on. Again, the engagement rate for Twitter over the course of the 12 weeks was 9.43%, which again is really, really high, seeing as the average for nonprofits is 0.055%. So I'm pretty happy about that. Another exciting thing about Twitter is we had the International Coral Reef Society reach out to us. They saw the work that we were doing. They reached out to us and they wanted to feature us on their page. So that was a cool way to, message out even more. And it was really exciting to show that, okay, someone's looking at us. For Instagram, I think Instagram was probably our most successful platform. I did 40 posts. There was a 345% increase in followers across the 12 weeks, 64 to 285. Again, there was that positive linear regression of likes. Comments were basically negligible. They didn't change that much over time. There weren't that many comments. And again, the engagement on average was about 17.88%, which is much higher than the average of 1.41%. And another really exciting thing about Instagram, exciting thing about Instagram is that we got a lot of direct messages from different individuals and different organizations, either just saying like, oh, I didn't know this, or this is really cool. And we got one woman reached out to us, said she was really passionate about this region. She's spent a lot of time there. She really wants to help out. We actually trained her, onboarded her, and she's running the social media now for the RSRF. So that was a really exciting thing to see that, someone was so inspired that they really wanted to take part of it. These are some of our most successful posts. I'm only going to go over just 2 of them, I believe. So the one on Twitter that was the most successful was a Valentine's Day post, and it read, it was this image here of the Turbinare reniformis. The caption read, once a year, at the end of November, the Turbinare reniformis releases all its gametes at once, hoping that over several days they will find a match in the water column. Talk about putting all your eggs and sperm in one basket. Hashtag Happy Valentine's Day. So I think that one was really successful because it utilized humor as a way to connect. It also was Valentine's Day. That hashtag probably got a lot more traffic because it was Valentine's Day. And it also employed that characterization of the coral. So we kind of made it we used a kind of traditional storytelling technique and made it like this own little character, like, oh, happy Valentine's Day from this little guy. And then the most popular post on Instagram was a video of Dr. Kleinhaus and a colleague working in the coral reef nursery in the Gulf of Aqaba. And it makes sense that the video was the most popular because basically videos are kind of taking over social media right now. TikTok, popularity of Instagram Reels, videos represent 75% of all global internet traffic, and that's just expected to just keep going up. So they're also just a really impactful way to bring somebody in. It kind of removes that barrier even more, like you're right there with the video. So that one was very successful as well. So overall, in terms of campaign successes, we had really high engagement across all platforms. We had increased followers across all platforms. That collaboration with the ICRS, we got the social media volunteer. Those are all successful. I believe out of the 4 goals that I started with, I think we hit 3 of them. So we hit, reaching building that broad base, spreading the scientific information, those sorts of things. The biggest weakness, I would say, is that we didn't receive any donations to the website. So that was a kind of a bummer. We did a donation call probably about once a week, maybe once every 2 weeks. And I think that was likely due to just low website traffic, and also there wasn't a lot of incentive to donate. We designed this certificate of appreciation for people who donated more than $17. They would get this sent in their email, but due to like technical difficulties, we couldn't get it up in time on the website. So we weren't able to use that, and maybe that would have helped, who knows, but that's something that the RSRF could definitely do in the future. I would have loved to have a couple more collaborations with different nonprofits. And I also think in the future we should do more videos as things are heading more towards that direction. So these are my recommendations that I developed through my research, through analyzing the successful and unsuccessful posts of my campaign. So basically it's what I've been saying: determine an audience early on and design all those materials with that audience in mind. Have clear measurable goals set before the start of the campaign and have a way to measure them, have like those metrics in your mind, a way to measure them and keep track of it. I think it's really important to focus on narrative, storytelling, and emotional connection. As we all learned through Carl's class, you can't change minds through facts, you have to change it through hearts, right? So you have to like kind of touch people in a different kind of way than just with facts. Also being transparent and connecting and personal with the audience. And people trust social media because it's their friends, right? It's their friends telling them this news, and it's their family. So you want to kind of be that for them. You want to find a way to be that credible source of information, but also not be hiding behind anything. Being consistent with your posts, setting a really clear schedule, finding the best time to post, which apparently is 8 a.m. to 12 p.m. on weekdays. Make it beautiful, memorable, and shareable. Obviously, people are interested in that. Capitalize on the trends, utilize videos, and have those incentives for donations. And I just want to say thanks to her, and thanks to Mark Lang for spreading the word. That's all. Questions?
We're running out of time.
Okay.
Great presentation, and we'll do questions very much.
Okay. So for my project this summer, I worked on creating the Long Island Beach and Water Quality mobile app. So first, before I started, I wanted to go over just a couple key terms so that we could all be on the same page, since I'm going to be talking about some technological things. So the first one, Chris talked a little bit about it in his presentation, but it's ArcGIS Pro, and GIS stands for Geographic Information System. And this mapping program allows you to visualize and explore and analyze data. You can create 2D maps, you can make 3D scenes, you can share your work online, and you can access data that other people have published. And we did some of that in our project. The next is ArcGIS Online, and this is basically a cloud where you can share your maps that you created in Pro online with other people, with an organization, or with the general public. And last, this is the big one.
is REST API, and this stands for Representational State Transfer Application Programming Interface. And APIs are essentially just a set of rules that define how applications can connect to and communicate with each other. So to put that more simply, it allows an application or a service to access resources from other applications or services. So it's almost like streaming data from one source to your project. For our work, we used REST APIs that we made to access data from the Department of Environmental Conservation and the Department of Health. And the main benefit to using REST APIs was that data would be automatically updated on our app when the DEC or the DOH made updates, rather than us manually updating our data. So to get a little more into the specifics of this project, it was led by Dr. Sunggil Jang of the Geospatial Center, and he advised the research assistants who were me. I was the lead research assistant. Jordan Russo, who is in the MCP program, was another research assistant. And then there was Renee Ward, and she's an undergrad geo student. The goal of this project was to develop a mobile app to present Long Island beach and water quality data by integrating different sources of information and then compiling them all into one place. This project, I joined in May, but the preparation for this project actually started in March. And the need for this project was due to the fact that water quality for Long Island's coastal areas comes from a variety of different sources and has never been spatially integrated for the public. This app will provide easily navigable location-based data for everyone to see in one simple app. And this can provide the public with an overall understanding of how Long Island's precious water quality resources are faring. So the first major task of this project was to obtain the various types of water quality data that we needed. So we used water quality index data from the Global Lab, beach closures data from the Department of Health, and shellfish closures from the DEC. So first, Dr. Christopher Gobler provided our team with an Excel spreadsheet of water quality index data from his lab. And then every week this summer, we've received updated data from their testing. So the Gobler Lab here at Stony Brook created this index, which provides the public with information about the current conditions of Long Island's coastal waters. And the index categorizes weekly water quality into 3 categories of good, fair, and poor, based on 6 parameters of measurements they take. And those are dissolved oxygen, average temperature, water quality, clarity, fecal coliform bacteria, chlorophyll, and harmful algae. Next, we acquired data for the beach closures from the New York State Department of Health Beach Water Quality website. And this website contains several JavaScripts in its actual website code that can present a web map that displays point features that indicate the positions of beaches in New York. And then these JavaScripts also contain beach closure data. So while the original plan was to use REST APIs to streamline this data to our app, isn't ArcGIS based, and they don't allow other web services to consume their content. So you can only see it on their website. So as a temporary workaround, Dr. Jang used a Python code that he wrote to extract data from the JavaScripts embedded in their website. So every day we run this code to get the most updated data, which is updated daily, and it essentially scrapes the data off their website that's hidden in the code. And then lastly, we had to gain access to the DEC shellfish closures data, which is available on their website. New York State regulates the use of and reports the sanitary conditions of shellfish lands. And the sanitary conditions of these lands determine if an area is certified or not, which indicates if shellfish can be taken for use as food. Regulatory closures are based on annual water quality analyses, and temporary closures can occur when an area that's normally open experiences a sudden and short-term degradation in water quality, like from stormwater runoff, harmful algal blooms, or biotoxins in the water. So to gain access to this, Jordan reached out to the DEC, and they shared their ArcGIS endpoint for this data via hyperlink to a REST API. So then the first step when we were getting into the actual app development was to create ArcGIS online web maps. So for each type of data, we created a separate web map and uploaded that to ArcGIS Online. And these web maps needed to be accessible to the public so they could actually view the data on our app, which presented us with one of our first problems for the water quality index data and beach closures data. So we had to figure out how we would be able to display the Gobler Lab water quality data so that the public would be able to view it on the map, but they wouldn't be able to actually go in and make any changes to the data. And then the second issue was how we can update the beach closures data if the Department of Health doesn't enable web-based sharing, because it would be completely unfeasible if every day we had to manually update over 200 beach data points. So to solve both these problems, we created REST APIs through a series of many steps in ArcGIS Pro and online. If I had like half an hour more of time, I would go over all of them. But to make it more simple, for the water quality data, we created one water quality data layer in Pro, which we then uploaded online. And then we created a new separate data layer that doesn't actually have any data in it, so the public wouldn't be able to change it, but it mirrors the data from the original layer so that the public would be able to see it. And this is essentially the REST API. We did a similar thing for the beach data. We recreated one data layer with beach closures information in it, and then a separate layer that mirrors these data points on the map. And then we published these online. But because we didn't have access to an Excel file like we did from the Global Lab, where we got the updates from every week, we have to run the Python script every day to open it and update it. And then in comparison, making the Shellfish closures data map was really easy. We just uploaded the data layer they shared with us from the REST API. Lastly, we wanted to display a map with all of this data together. So we just uploaded the two REST API layers and the DEC layer that they shared with us. And then the next step of this project was to actually begin developing our app in App Studio. And App Studio is a low-code platform, which means that users can develop applications more efficiently by reducing the amount of hand coding, which is when you write your own code that needs to be done. And the program provides app templates where you can customize the code to your own needs, but you can also create apps from scratch. And they also provide sample apps that perform certain functions like displaying your location on the map. So we did a mix of working with sample codes and then making our own codes. And App Studio uses QML, which is Qt modeling language, which is a declarative programming language that describes the aesthetics, like what the app would look like, and the behavior of a program's user interface. I'm going to skip this for time. It was just an example of what the code looks like. And now I'll actually give you an app tour of what this app would look like on your phone. So it opens up showing you our start screen, which is just welcoming you to the page, a brief description, and how to navigate it. Then when you open up, we'll take you to the web map displaying all of our data together. You can move around with your fingers, just like on Google Maps or Apple Maps. You can zoom in. So for this example, I decided to focus on Port Jeff Harbor. So if you click on one of the water quality symbols, you can see the name of it. This one's Port Jeff Harbor, the date of the last data, the class is good, and you can see more of the scientific information into what gives it this classification. If you wanted to check out what the beach was like near you, they're all labeled on the map, so you can scroll and click on it. This one's Bayberry Cove Beach. You can see it's open, and if you click the full history link, it will actually take you to the website for the New York State Department of Health, and you can view the full testing history to see, like, is this beach consistently good, or are there days where there's fecal coliform bacteria in the water? And then let's say you wanted to go clamming in Fort Jeff, so then you can scroll over to one of the shellfish lands, click on it, and this one's actually closed. And if you click details, it will take you directly to the DEC Shellfish Closures website, where you can see all the information that goes into this classification and further details. You can also close the legend that's in the top corner there if you didn't need to see all that information and make the screen more simple. You can switch maps. So let's say you just wanted to look at Global Lab water quality data. It will only show you that, simplify your viewing. You can see here, this is a red symbol, which means poor quality, and you can see the impairments are low dissolved oxygen, algal bloom, water quality, fecal coliform. So you can actually see what goes into the classification. If you click the I button, you can click report a concern to directly report any concerns at the beach to the proper authorities at the Department of Health. You can click feedback to take our survey that Jordan designed and tell us what you liked about our app, what you didn't like, and how we can improve it for the future. And then if you click the eye in the top corner, it will tell you more about our data sources. So if you click on those blue links, it will take you directly to their website. So this one is Chris Gobler's lab. So you can see our specific data than what we show on our app. It also tells you our team that developed it and data credits. Finally, if you click that location button, it will take you right to your location. So let's say you're near a beach, you could see all that relevant information right there on your screen. And that's the end of the tour. I think it's pretty cool. Future work on this app, we hope to improve the functionality and aesthetics, like creating a search bar where you could type in a specific address of a beach, like Cedar Beach, and it would take you there instead of searching for it visually on the map. And then another thing we want to do is create a home screen where you can see all of the options on our app at once. And I think that'd be an easier layout. We also want to expand the data collection to include other things like the location of harmful algal blooms, fish kills, and rip currents, which would be really convenient for like surfers and swimmers. Then that wraps it up. Hopefully this app is going to continue to be improved. I know there are plans to improve it even more and add those additional functions and features, and then officially released on the App Store next year. You can currently view it, the prototype that I just showed you, on App Studio Player, which is an app. We were also featured, which I thought was pretty cool, on some local news like Newsday and News 12. And I think once this app is finalized, it's going to be a really great resource for people here on Long Island. And then also quick shout out to our project funders, the Roth Foundation, Chicago Community Trust, and an anonymous supporter. That's it. Thank you.
You have a couple of minutes for questions.
Really nice. Sure. Sorry, what was it?
Sorry, did you have any GIS Pro this what it looks like?
Yes, we did some work in Pro, like the initial work of developing the data layers. So like we took scope. I didn't want to get into the details too much because it would take up so much time. But each week the Gobler Lab sends us an Excel spreadsheet, which is a table of all their data. Then we put that into Pro and we transform it from the table to actual XY points on a map based on the latitude, longitude coordinates they give us. Then we publish it to online. And that's what we did for the other types of data as well. So the foundational work was done in Pro, but then a lot of the other stuff was done online.
Another question? Okay, thank you. All right, so next, Siobhan.
Hello, everyone. I'm Siobhan Keeling, working on my capstone project, which is humpback whale entanglement in the New York Bight region. Dr. Leslie Thorne was my advisor on this project. So those of you unfamiliar with entanglement are probably wondering what it is. And so it's when fishing gear and line gets wrapped around the body of an animal, like as you can see in these pictures of these whales. And over time, with them driving it through the water, they become weaker and they can end up dying due to starvation since it inhibits their feeding, drowning because they're unable to surface, and finally infection from the line cutting into their body. Some whales are able to escape entanglement over time by shaking it free from their body or breaking it, and this leaves behind scars, which tells us that they've endured an entanglement event. So humpback whales are found around the globe. There's 14 distinct segments of them. The population that is part of my study is the West Indies one, which is number 1, and they end up migrating incredibly far distances all the way up to the Arctic. So more motivation behind this project, specifically looking at the region of the off the east coast of the United States in the Gulf of Maine region. On average, 6.5 whales die a year due to entanglement. And this might not seem like a large amount, but humpback whales and other whales have been, during the 20th century, had endured a lot of whaling, and so they ended up having their populations decimated. And so they're protected under the Marine Mammal Protection Act, as well as a moratorium on whaling. And unfortunately, we're inadvertently affecting them through entanglement and killing them. And so there's a potential biological removal threshold. And so if we go beyond this, their population will not be able to sustain themselves. And so in the Gulf of Maine, 57% of whales show entanglement scars, which means that a large proportion of whales are experiencing entanglements in their life. So areas that are prone to entanglement are the rostrum, pectoral fin, and fluke of the whale. So generally areas that can end up catching gear. And so I've included some diagrams on how this gear can get wrapped around a whale. And so the one on the far right, this isn't a humpback whale, this is a right whale, but gear gets wrapped around whales the same way. And so A shows how it can get wrapped around the rostrum. B shows how gear can get wrapped around their pectoral fin area and across their back. And then finally, this image on the left shows how gear can just completely immobilize a whale. So the objectives of my project were first identifying the presence of scarring. As you can see with these two whales on the bottom, whales endure a lot of scarring throughout their lifetime. Their skin ends up telling the story of what they've endured. And so scars appear light in dark areas of the whale and darker in the white areas of the whale. And so the next step of my project was determining whether or not it's from anthropogenic or natural sources. And then finally comparing juvenile versus adult scarring rate. So the methods of my project, drone footage was collected of 41 individuals from 2018 to 2021. I unfortunately have not been working on that project that long, but a member of my lab, Julia Stepanov, had been using drone footage to record the body condition of whales. And so she allowed me to use that footage to then take screenshots of the whales and then categorizing the scars I identified on those whales. And so in the top right, you can see we are deploying a drone off a vessel. And so the drone imagery took place in the New York Bight region, which is shown right here on the left. And so you're probably wondering why use a drone. And so in the study by RAMP in 2021, they recently discovered that a drone was able to capture more entanglement than previously identified through traditional methods, which is a photo ID of the fluke or observation off a vessel. So I want to look into that for my project since it is something recent. So you're probably wondering, how can you tell apart the scars? So starting off with anthropogenic scars, anthropogenic means from human impact. And so entanglement and vessel strike were the main things I was investigating. And so on the image on the top right, this is entanglement scarring on the fluke of the whale. And so a way you can kind of infer some things from entanglement is that that region kind of has a crescent shape where a rope has rubbed back and forth as the whale has swam. And you can also see that that's scarring on either side of the fluke on the bottom here with this region right here. It's the same one. And so you can also tell that this whale has sustained wrapping around the base of their tail. And so they have entanglement there. And then finally on the left here, this is from a vessel strike, and you can tell those apart based on kind of a spiral-like pattern where a propeller of a boat has repeatedly hit into the whale and removed chunks of its flesh. And so on another note, there's also natural scars that whales can endure. And so starting off with orca, the whale on the far right has some kind of rake-like marks that are like J-shaped. That's from an orca's teeth biting into the fluke of the whale. Their feeding behavior, so these two on the bottom show a bottom sideral feeding. So when a humpback whale goes to feed, they'll actually scrape their face along the bottom, creating scarring on like one region or the other. You can tell because that area is wider. And then finally, competition. Humpback whales get into a lot of stuff in their life. In particularly during the mating season, male humpback whales will fight each other for dominance and to escort a female, and they'll end up getting just random hatching straight line marks on their body, since their bodies have barnacles and it ends up biting into each other. So since the entire body of a whale can sustain a lot of scarring, I decided to divide the whale into 5 different regions, also to see which regions had the most entanglement. So these regions are the left rostrum, right rostrum, front, back, and fluke. Getting into the results of my study, there was an incredibly high amount of entanglement within the adult and juvenile populations. 86.7% of adults sustained entanglement in at least one of these 5 regions of their body, while juveniles, 61.5% of juveniles sustained entanglement in at least one region of their body. Interestingly, with juveniles and vessel strikes, 7.6% of juveniles sustained vessel strikes, while 0% of adults had any vessel strike scarring on their body. So looking at the specifics of body region entanglement, the fluke was a major region of entanglement with 73.3% of adults sustaining entanglement around their fluke, while juveniles had 57% of them having these marks. The next highest region is the back portion of the whale with 26.7% of adults and 7.6% of juveniles. The least entanglement scarring areas that I've seen in my study was the rostrum and the front of the whale. So something else I decided to look into, since I had this information, was the handedness scarring. So essentially the scarring of a humpback whale through bottom roll side feeding. So just like people, they kind of favor one side more than the other, like how people are right-handed or left-handed. And so humpback whales repeatedly roll on the same side to feed, and you can see by this left and right, and the majority of the whales were right-handed essentially. So with 53.6% of the whales in my study having the right-handed scarriness, 17% was left, and one whale actually had both the scarring on both sides of its rostrum. So I guess he's ambidextrous. And so that's something interesting to record. Also because 30 of the individuals had the scarring, 11 did not, and those were juveniles. So this is something that grows over time. And now I can measure this on these whales in this area. So broader impacts of my project. So first, 26% of entanglements were able to be identified through the use of a drone, since you wouldn't be able to identify them off a boat. This individual actually has entanglement scarring on the underside of its pectoral fins. And usually a way to tell that is there's kind of symmetry to it, and there isn't, like nothing would naturally make that scarred. And so, so my project supports that drone imagery is a useful tool in detecting entanglement rates. The next thing is that only juvenile sustained vessel strikes. And the New York Bight region is an incredibly busy area. And so it is affecting juvenile populations. And so that's something to look into further, because juveniles feed more inshore while adult humpback whales feed offshore. So we can kind of, focus on like what is causing this anthropogenic impact. So like either the fisheries in that area for entanglement, and for juveniles, the vessel strikes. And then finally, in my project, there was a high rate of entanglement, and most of the papers I read, they only focused on whales in general. They didn't divide them up into juveniles or adults. And they had about like a 57% to a 75% rate of entanglement. I looked at, and that's like all around the globe. There's like Australia and Iceland, but so my paper supports that entanglements are a growing issue. And now there's more methods on how to track it. And then finally, mitigation measures to things like going forward. So my paper has juvenile entanglement rates. That's not something that's looked into with a lot of different studies. And as I said earlier, juveniles feed more inshore. So we can look at those fisheries that end up impacting these species. And so, and the image in the top right, it shows a gill net, and so a humpback whale next to it. And that the buoy line is kind of the main area that humpback whales are being entangled in from what they've seen. And on the bottom here, you can see how a whale can get entangled in it. And so one of the first things that I had looked into was there's rope composition. And so a paper had found that having a rope with a kind of a more stretchier material would allow the rope to press against the skin of the whale instead of saw back and forth, creating the deep lacerations. And then another study had looked into the breaking strength of the rope. So if you decrease the strength of the rope, more whales would be able to break free out of entanglement, thus reducing the amount of entanglement that whales experience. And a lot of the papers had expressed that this would be investigated further for fisheries, but they don't think it would impact fisheries that greatly. And then finally, another alternative is ropeless fishing. And so using an acoustic transmitter to deploy a buoy rather than having a buoy at the surface would greatly reduce the amount of entanglement because there's less lining in the water. So finally, I'd like to thank the Thorn Lab, and particularly Julia Stepanek and Ellie Haywood. I worked with them closely, and without their help, this project wouldn't be accomplished, and also the support from all the members of my lab. That's my sources. Yes, so a lot of, I haven't looked into this specifically, but a lot of whales end up sustaining either infection, or if a line has stayed on a whale so long, it can actually amputate the limb. And so that is definitely a factor that ends up killing whales.
Analyzing sort of by hand all of these images must have been pretty difficult. So I was wondering if there was anyone developing more of an AI type of approach where you could, have a computer analyze the images for you.
So there is someone doing that already. They use like a computer identifier, but they found that it was kind of unreliable since it doesn't know how to detect which ones are from entanglement and which ones are just scarring in general. For my project, I actually had 2 sets of eyes look over the images I've taken from, since I would go through and take screen captures, and I had about 800 photos of these individuals. And so I got 2 sets of eyes to help me kind of either say yay or nay if it's an image or not.
They said there are bug strike scars on juveniles, but not adults. So how long does a scar last?
I don't know that, but I would believe since it's taken a substantial amount of its body out, I think it would survive its entire lifetime. Like it would last.
Right, I would think so too. So adults should have scars that they acquired as juveniles.
Right.
That seems odd.
Yeah. That is very odd. I know that the 41 individuals that have come through the New York Bight region isn't the entire population. So there could be some adult whales that actually forage not in the New York Bight region and avoid that area entirely so they don't encounter vessels.
How do you classify a whale as an adult? How big does it have to be?
I don't know the exact body size. My lab mate Julia Stepanov had looked into it, but there is a specific measurement of the size of the whale. I don't know it all. Any other questions?
Almost right. Timing.
Alrighty. If there's no questions, and I think that's it.
Next up is Jordan Russo. I just want to say this is the most beautiful color scheme I've ever seen.
Thank you.
For a shark presentation.
That's blood in the water.
No, it's pink. It's pink.
All right, so this summer I worked for my capstone with my advisor, Dr. Michael Frisk, and his lab to build and deploy 3 real-time monitoring systems off of the coast of the South Shore of Long Island. So with the increasing number of sharks returning to the waters off of the coast of Long Island in the summer months, it's really important to avoid negative interactions between the fish and with beachgoers. So to do so, this real-time system will immediately alert us via text and email whenever a tagged fish swims within 400 to 500 meters of any of the systems. This real-time array will help provide information that's important for protecting sharks through alerting management agencies of their presence to avoid incidental capture and also, again, to inform beachgoers. Unfortunately, due to delays in shipping of some of the parts, the real-time deployment has been delayed until further notice. However, I was able to gain experience with the acoustic telemetry, the technology behind the real-time array, animal protocols when shark fishing, and the maintenance of the current acoustic array network that's operated by the Frisk Lab. So the need for this project comes in 2 forms: conservation and ecological importance. In terms of ecological importance, shark movements and population abundance reflect the overall health of the ocean. Sharks are a great indicator of the ocean's general health and the ecosystem's maturity. And as apex predators, they assert top-down control over lower trophic systems in the ecosystem. So if sharks were to be removed from the ecosystem, the important energy pathways between the lower trophic levels would be diminished. And this reduction of shark biomass has been associated with the decline in ecosystem maturity and the resilience here in the Great South Bay. And in other systems, it's shown that the elimination of top predators has led to an algae-dominated system. Globally, many sharks have declined, with numerous of their species being listed as endangered, threatened, or of concern on the IUCN Red List. And this is very different compared to their historical populations of sharks, which were much higher than they were now. And if and as their populations do return to their historic abundance, the conservation and management efforts are going to be a lot more difficult to deal with increased human population. So overall, an ecological understanding of the shark movements can help better develop management and conservation efforts for their populations. This study aims to assess the practicality of using these real-time acoustic telemetry to monitor and characterize the nearshore movements of coastal sharks. So again, 3 real-time receiver stations are going to be deployed. 2 of the stations are going to be off the southeastern end of Long Island, 1 at Fitch Plains and 1 off Montauk Point. And the other one is going to be south of the Jones Inlet. These locations were selected based on human activity, so beachgoers and surfers, and was also combined with the probability of detecting white sharks, as well as other coastal shark species of interest that have been acoustically tagged and detected on the existing coastal array system. Each of these receiver stations are going to be deployed half a kilometer from the shoreline to avoid potential interactions with commercial fishing vessels that have been known to operate around these identified locations. So again, for this project, 3 buoys were bought. They are from the company Nexsen, so they're Nexsen CB150 buoys. These buoys have adequate topside space to accommodate various telemetry modules, including cellular, GPS, and Wi-Fi. And the underside of the buoy includes a stainless steel instrument cage that secures an RX Live cabled receiver. These receivers work with multiple frequencies, including 69 kilohertz tags, which are the most common type of acoustic tag. This is a picture of the buoy once we put it all together yesterday in the boathouse. So again, while working on my capstone project, I was also able to gain firsthand experience in shark tagging and the animal protocols. A majority of the shark fishing occurred in the summer months at several locations, including the Rota Wreck, which is a small shipwreck in New York State waters. That's approximately 1 kilometer off Gilgo Beach. And the other location was the Oregon Wreck, which is a larger shipwreck located in federal waters, about 15 kilometers south of Fire Island. So after we hook a shark by rod and reel, phase 1 of the shark field sampling begins with morphometrics, which are length and girth. The 3 length measurements are precaudal length, fork length, and total length. And the 5 girth measurements are frontal, lateral, proximal, caudal keel, and the general circumference, which gets measured around the fattest area of the shark. We then take a muscle biopsy sample by making a small incision at the base of the dorsal fin and using a small melon boss scooper to collect 3 to 4 muscle samples. And in the lab, these muscle samples are used for mercury analysis and stable isotope analysis. A 1 centimeter fin clip is also collected from the trailing edge of the dorsal fin. And finally, a predator program tag is placed under the trailing edge of the dorsal fin. This predator program tab is a cooperative program run by NOAA, and it allows recreational anglers, the commercial fishing industry, and NOAA fisheries to learn more about the life history of sharks in the North Atlantic. In the past, this data from the tagging and the recapturing of sharks has helped the scientists collect data on growth, movement, and capture rates of Atlantic sharks. Then phase 2 of shark sampling begins when we roll the shark upside down, allowing the shark to enter a state called tonic immobility, which is a temporary state of inactivity. Fecal and cloacal swabs are collected and used to characterize the microbiome. These samples are also used to look at the diet composition as well as microplastics. Diet is also referred to as like a key that can help unlock critical information about how sharks are connected to their habitats, such as how and why they use certain areas, where they go, and their overall food web interactions. A blood sample then gets collected from the shark. These samples are spun down and separated into plasma and packed red blood cells. The plasma is used for stable isotope analysis and fatty acid profiles. The last step is the acoustic tag surgery. So using a scalpel, a small incision is made and a Vemco V16 acoustic tag is placed in the column and then sutured up. And then after that, we release the shark, which I have a video of, when we roll it back into ventral recumbency and slowly hold the tail as it gets ready to swim. So with the existing coastal array by the Frisk Lab, it's used to track the movements of Atlantic sturgeon, as well as other important finfish and elasmobranchs. So in 2010, 4 acoustic receiver arrays were deployed and arranged perpendicular to the shore in Montauk, Fire Island, Jones Beach, and the Rockaways. And in 2019, this project was then expanded with the addition of array networks on the Atlantic Beach, Fire Island, Reynolds Channel, Elder Meadow Island, Swift Creek, and the Meadowbrook State Parkway bridges, all of these which are located on the major inlets to the Great South Bay. With this array, a total of 248 elasmobranchs have been tagged, and of these, 82 have been detected on this coastal nearshore array. The most abundant shark species tagged and detected are dusky sharks, blacktip sharks, sand tiger sharks, sandbar sharks, smooth dogfish, spiny dogfish, and the common thresher. From this data, detection patterns revealed that these elasmobranchs typically enter the state waters of New York at the beginning of July and remain until the early fall around September. Only focusing on the species of interest, the sand tiger, common thresher, and sandbar sharks were typically detected from the Rockaways to Fire Island, with very few detections of these species on the Montauk array. And it's speculated that this could be due to where they have been tagged. However, all species show residency in the coastal array, suggesting that this region provides an important habitat and likely supports important prey species during the warm summer months. Rate of movement estimates for all elasmobranchs were typically slow, suggesting that the New York Bight region provides sort of a stopover habitat for some individuals. Yet previously studies on species such as sand tiger sharks suggest that some individuals migrate as far north as coastal Massachusetts, and therefore coastal New York can serve as both a migration corridor as well as a critical habitat. So overall, this previous project illustrates that there's a need for future work to increase the tagging efforts and the array coverage in the more eastern areas of New York. The majority, again, of the detections were concentrated between the Rockaway array and the Fire Island array, with a lot of fewer detections on the Montauk array. This relates to the additional deployment of the 3 real-time arrays, as 2 of these 3 systems are going to be deployed onto Montauk. This is just some GIS data of some tagged sandbar sharks that were detected on the coastal array in New York state waters between summer of 2018 and 2019. This is movement of 7 individual sand tiger sharks that were detected on the coastal array in New York. And as you can see on the top one, they were detected all the way down in Cape May, New Jersey. And this is just the movement of 3 individual common thresher sharks that were tagged off the coast of New York and detected. So to kind of sum everything up, within the past few weeks, I know I myself have received several notifications of shark sightings and beach closures on New York. This has prompted local officials to close several of their beaches and send police officers out in helicopters, in boats, and on jet skis to shark patrol. An interesting quote from Wildlife Conservation Society's Han Walters says that people are seeing sharks because there are more people with more smartphones and more drones and more social media. So in other words, more people are looking for them. However, if the shark numbers are actually increasing off the coast of New York and New Jersey, it would actually be a good thing. It would show that this species and these animals are recovering from severe overfishing and even pollution. One way to be able to see if these shark populations are increasing is through the use of the existing coastal array, as well as the addition of the new real-time array. With the real-time array, again, we do get notified instantly via text message and email if any acoustically tagged fish swims within 400 to 500 meters of the buoys. This information will also be used to plot habitat preferences, migrations, and a lot more. With the real-time array, the instantaneous alerts will allow us to have a better window of shark presence in the waters near Long Island. And these windows could be used to enforce areas such as closed areas where no fishing is going to be prohibited, as a way to help reduce shark mortality. Specifically in New York, closed areas are really important for certain species such as sandbar, dusky, and sand tiger sharks due to their life history traits. And this real-time array can also help us avoid the negative interactions between the beachgoers and the sharks, in addition to management, mitigation, and conservation efforts for sharks off the coast of Long Island. That's it.
Couple of minutes for questions.
How long does it take you to do all of this different things?
It kind of depends. Most times we get them in and out in 15 minutes max, maybe. Kind of a time crunch. Yes.
Is there any estimate on when you'll get the stuff for the release?
Last I heard, we were waiting for specific wire that was supposed to be here a few weeks ago. So I think it should be here, but we're working on an Orsted project right now. So we've been a little backed up.
So what are indications are there of changes in the shark population?
I mean, alongside just more people seeing more sightings, there's been a lot, but even with data on the current coastal array, it's kind of hard because we go out every few months and we have to retrieve the data. With this data, we'll be able to get it instantly to kind of better see how many are here and when they're here.
There hasn't been a recent assessment by the fisheries service of shark population.
And I'm not sure.
No one knows. No one in the room knows. Anything else? I think we're actually only down to a few seconds. So, okay. Well,